So, ladies and gentlemen, may I have your attention, please? The opening ceremony of AIRIS 2025 will begin very soon. So please make your way into the hall and take your seat. Also, please set your mobiles or other electronic devices to silent mode. The simultaneous interpretation service is available. Please note that channel 1 is for Korean and channel 2 is for English. 잠시 후 AIRIS 2025 개막식 시작하겠습니다. 여러분 착석해 주십시오. 본 심포지움 동시 통역 제공해 드립니다. 통역이 필요하신 분께서는 수신계 채널 고정해 주십시오. 한국어는 1번, 영어는 2번 채널입니다. 잠시 후에 오프닝 영상과 오프닝 퍼포먼스가 진행이 될 텐데요. 이 홀이 암전이 됩니다. 여러분들 가급적 이동을 삼가해 주시면 대단히 감사하겠습니다. In a few minutes, the opening video and opening performance, the light of this hall will be blocked out. It will be turned off. So please, for your safety, please avoid the moving to other table. Thank you. Wow, the performance team 생동감, ladies and gentlemen, please give another round of applause. Thank you. The Korean traditional dance converged with the digital media technology present the truly captured exciting moment of AIRIS 2025. Ladies and gentlemen, did you enjoy it? Thank you. I hope you did. Distinguished guests, ladies and gentlemen, good morning. Welcome. And thank you for your presence at the opening ceremony of AIRIS 2025. I am Lee Song-hye, and I'll be your MC for today. It's a great honor and pleasure to have you here for such a meaningful and a wonderful event. We'd like to express our sincere gratitude to all representatives from each regulatory authorities and leading experts from academia industry with us today. So I hope you have a very meaningful and fruitful time during this symposium. To begin, I'd like to introduce our guests of honor who grace this occasion. So when each name is called, please welcome them with a warm round of applause. Her Excellency Yoo Kyung-oh, Minister of Food and Drug Safety, Republic of Korea. Dr. Seok Kang, Director General of National Institute of Food and Drug Safety Evaluation. Dr. Ki Dong-park, Medical Officer, Division of Health System and Services, WHO. Samir Pusari, Lead AI, Frontier Technologies, WHO. His Excellency Dr. Ali Mohammad Gamlawi, Minister and Chairman of Egyptian Drug Authority. Professor Mojisola Christian Adeye, Director General of NAFDAC, Nigeria. And this time, let me introduce the 4 esteemed chairmen of associations in Korea. So at the conclusion of the introduction, please give them a round of applause. Mr. Yeon Pong-no, Chairman of Korea Pharmaceutical and Biopharma Manufacturers Association. Mr. Young-min Kim, Chairman of Korea Medical Devices Industry Association. And Mr. Seung-hyun Son, the President of Telecommunications Technology Association. And Mr. Jae-mo Han, the President of Korea Dental Devices Industry Association. And lastly, we are honored to represent each country's regulatory authorities, leading experts, diplomatic corps, industry, and corporation. Would you please welcome each other with a big round of applause? Welcome to AIRIS. Thank you very much. Before the opening ceremony, there will be an exhibition tour, so you will witness the AI technology and status of the convergence and digital in technology and innovation. I hope you have a very meaningful time during the exhibition tour. To mark the beginning of this event, I'd like to invite Honorable Yoo Kyung-oh, Minister of Food and Drug Safety, Republic of Korea. Ladies and gentlemen, please welcome Minister with a big round of applause.
안녕하십니까. 대한민국 식품의약품안전처장 오유경입니다. 오늘 AIRIS 2025를 축하해 주시는 대한민국 김민석 국무총리님, WHO 테워드로스 아드하눔 거브로이어수스 사무총장님, 이집트 의약품청 알리 모하메드 감라위 장관님, 나이지리아 식약청 모지솔라 크리스티 아데예예 청장님, 덴마크 의약품청 닐크 파크 비에가르드 청장님께 감사드립니다. 또한 오늘 세계 각국에서 AIRIS 2025에 먼 걸음 해주신 모든 분들 환영합니다. 세계는 지금 인공지능 대전환의 거대한 물결 속에 있습니다. 의약품과 의료기기 분야에서도 진단과 수술의 보조, 후보 의약품의 발굴, 제조, 임상시험, 시판 후 안전관리까지 전 주기적으로 인공지능이 활용되고 있습니다. 대한민국 식품의약품안전처는 세계 최초로 디지털 의료제품법을 제정하였고 생성형 AI 의료기기 허가심사 가이드라인을 올해 발간하였습니다. 또한 AI 기술을 활용한 규제 체계에도 앞장서고 있습니다. 허가 심사 단계에서 방대한 허가 자료의 심사에 AI를 활용하는 AI 심사관 제도의 도입을 추진 중입니다. AIRIS 2025에서는 규제기관, 학계, 산업계 최고 전문가들이 모여서 인공지능 혁신의 사례를 공유하게 됩니다. AIRIS 2025가 의약품, 의료기기 분야에서 AI가 안전하게 사용될 수 있도록 새롭고도 보편적인 국제 규제의 틀에서 소통하는 글로벌 플랫폼으로 지속적으로 발전하기를 기대합니다. Welcome to AIRIS 2025.
Thank you. Minister gave us the opening remarks. She emphasized that the international community must join forces and engage in dialogue and establish universal regulations that can be applied globally. Thank you for sharing the vision, and that unites us. Thank you very much. To encourage the minister and the organizing committee who put a great effort to make this event possible, please give another round of applause. This time, we are honored to present the welcome address of Dr. Tedros Adhanom Ghebreyesus, Director General of WHO, co-host of this symposium. He could not be here with us today, but he has kindly sent a video message for us. So please turn your attention to the screen.
Honorable Minister Oh Yu Kyung, Excellencies, dear colleagues and friends, WHO is pleased to co-host this symposium together with the Ministry of Food and Drug Safety of the Republic of Korea. We're grateful to our partners for their leadership and delighted to welcome all of you here in Incheon. Artificial intelligence is rapidly reshaping our world, including the development and regulation of medical products. As AI becomes more sophisticated and its health applications expand, so must our efforts to make them safe, effective, ethical, and equitable. This symposium builds on the work of the Global Initiative on AI for Health, a joint effort by WHO, ITU, and WIPO. It offers an important forum for regulators, researchers, innovators, and industry to share experiences and to chart pathways for the regulation of AI in medical products. Building on the success of last year's event and WHO's guidance on regulatory considerations for AI in health, this symposium will explore critical developments shaping our future. This includes the use of digital twins, large language models, responsible innovation, and real-world implementation challenges. I thank you for your commitment to ensuring that safe and equitable AI empowers health workers and improves the health and well-being of all people everywhere. Kamsahamnida. I thank you.
Thank you. 감사합니다. Dr. John Olgebriosus gave us a warm welcoming speech. The message reminds us the importance of our responsibility and commitment in AI era. And also he wished the symposium will be a platform for sharing experiences and shaping the future together. Thank you. And next we will hear congratulatory speeches from our esteemed guests from home and abroad. And first of all, we are delighted to present a video message from Prime Minister Min Seok-kim of Republic of Korea. Let us watch the screen.
안녕하십니까. 국무총리 김민석입니다. 식품의약품안전처와 세계보건기구가 공동 주최하는 AIRIS 2025 개막을 축하드립니다. 잠시 후 기조연설을 해주실 데이비드 베이커 교수님을 비롯한 모든 참석자 여러분들을 진심으로 환영합니다. 하루가 다르게 발전하는 인공지능 기술을 빠르게 도입하고 효율적으로 활용하는 것은 모든 산업 분야의 핵심 과제입니다. 의료 제품은 인공지능의 효용이 특히 큰 분야입니다. 인공지능을 활용한 방대한 데이터 분석은 신약 개발 비용과 기간을 획기적으로 줄이고 있습니다. 임상시험 성공 가능성도 빠르게 높아지고 있습니다. 경제적으로 디지털 헬스 산업은 연평균 25%씩 커가는 미래형 성장 산업입니다. AI 기술을 의료 제품 분야에 안전하고 효율적으로 활용하기 위해서는 규제의 기준과 방향 역시 새로운 기술에 맞도록 빠르게 정비되어야 합니다. 이를 위해서 정부, 학계, 산업계가 함께 노력해야 합니다. 대한민국 정부는 AI 의료의 지속가능한 발전과 혁신을 위해 세계보건기구를 비롯한 국제기구 및 각국 정부와 협력을 확대하고 있습니다. 이번 심포지움의 논의를 통해 글로벌 AI 의료 제품의 지속가능한 발전을 위한 협력의 기반이 강화되기를 기대합니다. 감사합니다.
Thank you, Prime Minister. Prime Minister remarked that the government of the Republic of Korea is strengthening cooperation with the WHO and other international partners to strengthen and foster growth in AI healthcare. And also he wished the symposium would enhance the solidarity and cooperation which all around the world. Thank you for encouragement. And next, I'd like to invite His Excellency Dr. Ali Gamlawi, the Minister and Chairman of Egyptian Drug Authority, to deliver congratulatory speech. Please join me in welcoming him with a warm round of applause.
Good morning. Your Excellency, Minister Oyu Kyong, my friend and colleague from Africa, Professor Muji, Director General of the National Agency for Food and Drug Administration and Control, NAFDAC, Nigeria, esteemed leaders from WHO and national regulatory authorities, distinguished guests and colleagues from industry and academia, good morning. It's a true pleasure and honor to join you today at this remarkable gathering, co-hosted by MFDS and WHO, a forum designed to help our global community explore how artificial intelligence can reshape the medical product life cycle safely, ethically, and effectively. Egypt and South Korea share a long-standing relationship built on mutual respect and shared ambitions. Today, we stand closer than ever, united not only for our history of cooperation, but also by our common vision for the future of pharmaceutical innovations. In this spirit, allow me to begin by extending my warmest congratulations to MFDS on its recent designation as a WHO-listed authority across all regulatory functions. These achievements, announced in August 2025, crowns years of consistent excellence and positions MFDS at the forefront of the global trust and reliance. It's not only a recognition, it's a catalyst for broader access, stronger partnership, and deeper collaboration extending for beyond South Korea's borders. Congratulations. Chow Hao Yu. I hope I'm saying it in the right way. This is a congratulation we bring to you from the land whose enduring legacy in medicine and pharmacy has inspired generations. The timeless land of civilization, Egypt. More than 7,000 years ago, ancient Egyptians produced what is widely regarded as the world's first written pharmaceutical guidelines, the AIRIS papyrus. I don't know if you heard about that. It's one of the papyrus which has been founded more than 3,500 years ago by a German Egyptologist, and his name is George AIRIS. This papyrus, stretching over 20 meters, it contained more than 800 prescriptions spanning dermatology, gynecology, birth control, dentistry, internal medicine, and beyond. And is still well kept at Leipzig University in Germany. I encourage you to Google it. And this document was not merely a record of science. It laid the foundation for pharmacy as we know it today. Fast forward to the present, Egypt's pharmaceutical sector has become one of the most dynamic in Africa and Middle East. We host more than 179 pharmaceutical factories, 150 medical device manufacturing sites, the same for cosmetics, and 4 biologics facilities and 5 API plants. Our local industry covers 91% of national pharmaceutical needs, which means that out of every 100 packs we produce locally. We sell in Egypt, we produce it locally. The pharmaceutical market alone is valued more than $6.5 billion US. Together with medical device and cosmetics, this can reach more than $12 billion US. Together, these factors have made Egypt the largest pharmaceutical market in Africa, with more than 27% of the entire continent. These figures underscore a simple truth. Egypt is not only your geography closest point in Africa, it's also your most practical gateway to the continent. We warmly welcome South Korean and global investors and partners to join this journey. With our strategic location and rapidly expanding market, Egypt offers unparalleled opportunity, providing access not only to our 110 million inhabitants, but also for the 1.4 billion inhabitants living in Africa. Of course, opportunity means little without trust. This is why I'm proud to share that the Egyptian Drug Authority is the first and still the only national regulatory authority in Africa to achieve WHO maturity level 3 in both medicine and vaccine producing. This recognition is a testament to our uncompromising commitment to quality, safety, and efficacy. For us, growth never comes at the expense of standards. Once again, I'm deeply grateful for this opportunity to join you at Artificial Intelligence Race 2025. It's a true honor to stand alongside you as a colleagues, partner in shaping a future when innovation and humanity walk hand in hand. Thank you very much.
His Excellency Dr. Ali Gamlawi, Minister and Chairman of EDA, gave us a warm the congratulatory speech. He Minister remarked that on the importance of international cooperation and need to align the regulatory standard with the emerging tech, including AI. Also, we came to know that Egypt is the gateway to the global market of the pharmaceutical industry, and Egypt is welcoming all of you. Thank you very much. And this time, let me invite Mojisola Cristiano Adeye, Director General of NAFDAC Nigeria, who will deliver congratulatory speech. Please welcome Director General Adeye.
Good morning, everybody. Your Excellencies, Honorable Minister O. Yonkun, distinguished colleagues from the Ministry of Food and Drug Safety, leaders and experts from the World Health Organization, fellow regulators, and heads of agencies, good morning. It is an honor to be here at AIRIS 2025, a gathering that reminds us of why we do this work: to make sure medical products are safe, effective, and trustworthy in a world where technology is moving faster than ever. On behalf of NAFDAC, I amplify our heartfelt congratulations to the Republic of Korea on its remarkable achievement of attaining WHO-listed authority or WLA status. Recently, the World Health Organization affirmed that the Ministry of Food and Drug Safety of Korea has attained the enviable feat of WHO-listed authority across the two scopes of medicines and vaccine regulation. This means MFDS is a regulatory authority which has been documented to comply with all the relevant indicators, and there are many relevant indicators from WHO, and requirements specified by WHO for the two scope based on an established benchmarking and a performance evaluation process, very tedious process. It shows that MFDS has reached the very highest level of performance in overseeing medicines, vaccines in all the 8 regulatory functions. In short, it is one of the regulators in the world that the global community can fully rely on for decisions that protect public health. I can say with certainty that the WLA achievement for MFDS is no small feat. It takes years of building strong systems, digital tools, and a culture of transparency. Republic of Korea has not just passed a test, it has set a new global standard. I heartily congratulate you and your indefatigable MFDS staff on this great achievement. This declaration validates the dedication, sacrifices, rigor, and sleepless nights of your dedicated staff, and indeed, years spent towards institutionalizing systems, processes, and structures that guarantee safe and quality-assured medicines and vaccines. Congratulations. Why is this recognition matters? Why is this so important? By being recognized as WHO-listed authority, Korea's regulatory decisions can now be trusted and relied upon by other countries. This makes it faster and safer to bring quality medicines and technologies to patients everywhere. It also strengthens our ability to work together when new opportunities like AI-driven regulation in drug development, drug approval, quality, safety, and efficacy emerge. In Nigeria, our agency, NAFDAC, has also been on this path. So I know what MFDS has gone through, mostly. In June this year, NAFDAC successfully maintained WHO maturity level 3 for the regulations of medicines and vaccines. It's the first regulatory authority in Africa to maintain WHO ML3. This means our system is stable, well-functioning, and reliable. But we are not stopping there. Our aspiration is to move to maturity level 4 and then to WLA, just as Korea has done. And with the MOU that we have signed together, it makes it easier. We want to join the community of regulators whose decisions others can rely on with confidence. This week, here at AIRIS, we have an opportunity not only to celebrate MFDS recognition, but to translate it into shared progress. To translate it into shared progress. I propose this together, that we develop a practical playbook for how to regulate AI-enabled health products, build protocol for reliance so that LMIC regulators can benefit from WLA agency decisions, create digital commons where regulators can share tools and test data, establish skills exchange program to train the next generation of assessors, and set up a joint vigilance network to quickly share safety signals across borders. Colleagues, MFDS achievement is more than a milestone. It is a message that with persistence, investment, and partnership, regulators can rise to meet the challenges of a changing world. And our thanks go to WHO. NAFDAC is inspired by this progress. We commit to deepening our collaboration with MFDS, WHO, and all partners here, so that one day, soon, Nigeria too will stand among the WHO-listed authorities. Together, let us ensure that every patient everywhere can trust the medicines and vaccines or technologies they receive. Congratulations again to Republic of Korea's MFDS, and thank you, WHO and AIRIS, for bringing us together for this vital conversation. Thank you very much.
Thank you. Mojisola Christina Adeye, Director General, gave us a congratulatory speech. She emphasized all of our cooperation between regulators will create a huge achievement. Thank you for words of encouragement. To commemorate this occasion, now we will proceed with a group photo session. So I'd like to invite all the guests of honor in the front row. May I invite you to come up to the stage for photo session? 네, 기념사진 촬영을 위해서 맨 첫 줄에 앉아 계신 내빈분들 잠시 무대로 모시도록 하겠습니다. 곧이어 기조 강연이 있기 때문에 참석자분들께서는 자리에서 잠시만 기다려 주시면 감사하겠습니다. After the photo session, we will proceed the keynote speech right away. So we kindly ask our participants to be seated and wait for a moment. Thank you. Thank you. Would you please look at the camera in the middle? All of your smile are so much beautiful. Would you please just step down, the one step down? All guests of honor. Yes. Thank you. The regulation for AI Together for Tomorrow, we collect our vision that unite us. To commemorate this occasion now, we will take a commemorative photograph. So first, please give a big smile for the camera, the center. Yeah, would you please, yes, a little wider? Thank you. Thank you. Please give a big smile. AIRIS 2025. 여러분 박수로 함께 해주십시오. And this time, do you know the heart cross, a little heart cross to symbolize love and peace around the world? Please make the heart cross with your fingers and I love AIRIS. Okay, I love AIRIS. Thank you. And for the success of AIRIS, please give a big hand. Thank you. Thank you very much. Thank you for your cooperation. So you may return to your seats as we proceed with our keynote speech. Thank you. Now we will now begin our keynote speech. It's our great privilege to introduce Dr. David Baker, the 2025 Nobel Laureate in Chemistry, professor at University of Washington and director of Institute for Protein Design. He will deliver his keynote titled Design of New Protein Functions Using Deep Learning. This keynote will be delivered through a pre-recorded video, and the Q&A session will be conducted live with our moderator. So I'd like to invite Professor Min-kyung Baek from Seoul National University. She will be our moderator. So ladies and gentlemen, please welcome our moderator to the stage. Thank you. Now, let's meet our keynote speaker, Dr. David Baker, on the screen.
Today, I'm excited to share with you some of our recent work on the design of new protein functions using deep learning. So the proteins in our bodies and in all living things evolved to solve life's most complex challenges. In the last 5 years, we've developed powerful AI tools for generating new proteins with new functions. And this is what I want to tell you about today. So protein design is very distinct from protein structure prediction. So in protein structure prediction, we start with biological information, the sequences of proteins in nature. We start, in fact, with the genes in our genomes and in the genomes of all living things. We translate those into amino acid sequences, and then from the amino acid sequences, we predict the structure of the proteins, and from those predicted structures, inferences can often be made about biological function. And this is the problem that AlphaFold from DeepMind made huge progress towards solving. So protein design goes in the opposite direction. The flow of information is in the opposite direction. We start with a desired function that doesn't exist. We make up a protein that has this function that we predict will have the function. We design an amino acid sequence that is predicted to fold to that structure. And then we work backwards to using the genetic code in reverse to identify a DNA sequence which will encode this protein. The important part, or an important part, is that we then have to synthesize this gene, make a synthetic piece of DNA, because it's a brand new protein, so it doesn't exist anywhere in nature. So we synthesize a gene encoding the new designed protein. We put it into bacteria or some other organism. The bacteria then produce the protein, and we extract it from the bacteria, and we determine whether it does what we designed it to do. So whereas in structure prediction, there's not really an experimental step, because you start with DNA sequence or amino acid sequence, and you go to three-dimensional structure and function. In the case of protein design, the final experimental step is very important, because that's when you know whether or not the sequence you've designed it actually has the function you intended. So we have, as I said, been developing deep learning methods to solve this problem. And the most powerful of these methods are diffusion methods, and they're inspired by deep learning methods for generating images, where you take images off the internet or some other source, you noise them to different extents, and then you remove the noise. You train a network to remove the noise. And once this has been done, you can start with completely random noise and then progressively denoise it to generate images that look like they came from your training set, but they're in fact completely new. We did the same thing with proteins. We took all the protein structures in the PDB, we noised them to different extents, and then trained a network to remove the noise. And once we had done this, we could start with completely random distributions of residues, progressively denoise these to yield structures that look like they could be real protein structures, but in fact didn't exist before. Well, just like in the image case, where it's not very useful to generate images in an unconstrained fashion, it's not very useful in the protein case just to generate proteins with no function. So the important point in both cases is how to condition these methods so that they generate what you want them to generate. So in the top case, that could be a text prompt, generate an image of a cat sitting on a table. In the bottom case, we can condition this diffusion process in several ways. One way is to provide the structure of a target, here the insulin receptor. And then the diffusion process, by progressively denoising the random cloud of residues in the presence of the target, generates a protein that looks like it should bind the insulin receptor. It basically looks like a protein that bound the insulin receptor would look like if it were in the PDB. And so then, as I said, we can make the protein in the lab and determine whether it has the desired function. So we have been using this approach to make design proteins against many different targets. So here on the left, you see a diffusion trajectory aimed at the TNF receptor, which plays a major role in inflammation. When we make the protein, we find it binds to the TNF receptor with extremely high affinity, single-digit picomolar, and it's very effective in treating inflammation in animal models. We can guide the diffusion process, or we can condition it by also telling it what sort of structure we want. So in particular, we can say, generate an antibody-like scaffold, in this case, a nanobody, or we can say, generate a single chain and FC, and FE. And in this case, the method generates a protein structure that binds the target, but also has the desired fold, in this case, an antibody fold. And since the method has been trained on antibodies, among many other things, it knows that it should use the CDRs, it should generate CDRs which match against the structure of the target. And that's what you see here. This is actually a comparison of the cryo-EM structure of this design to the design model, and you can see they're very, very close. So this is exciting because antibodies are obviously of great interest in biotechnology currently and in pharma, and being able to design them on the computer provides a new way forward for this important area. We can, as I said, we can use this approach to generate SCFVs, which have 6 CDRs from what was originally a heavy chain and a light chain. In the SCFV case, they're just one continuous chain. Now, in the case of actual antibodies, one discovers an antibody, say by immunizing an animal, and then generates an SCFV by connecting the heavy and light chain variable regions. In this case, we just start by generating single chain, but we can go backwards to generate antibodies. That's what you see in the middle here are these quite reasonable affinity antibodies, which have been generated by first designing SCFVs and then fusing them, decoupling the heavy and light chains and putting them onto the FC framework. This shows that these structures can have the CDRs can be designed with very high accuracy. You see a superposition of the CDRs and the experimental and structurally determined CDRs at the bottom here. So another very important class of molecule in the immune system are the MHC. The MHCs display peptides to be recognized by T cells, by the TCR. And so they present, for example, neoantigens. And so if you want to detect cancer cells based on differences on the inside of the cell, your best bet is to target the peptides from that cell in complex with the MHCs on their surface. And so here you see a diffusion trajectory that's generating a binder that lies across the surface of the peptide MHC complex. And when we design these, we focus on recognizing the amino acids that stick up from the peptide rather than making extensive interactions with the MHC, because we really want these to be peptide specific. This on the right is a very famous case where a T cell therapy was made against the MAGE peptide, but the patient ended up dying because there was also cross-reactivity against Titin. Here, using this approach, we can design binders that are specific for MAGE and show no reaction, cross-reactivity with Titin. And the crystal structures of these designs can be extremely close, or where we've solved them are extremely close to the design model. Here you can see it's very hard to tell the difference between the predicted structure of the design model of the peptide MHC complex and in complex with the binder, and the binder's up here. Here's the peptide. And the crystal structure, and you can see they're the side chains are very, very close. We can also use these methods to design antagonists and agonists for GPCRs. Here are examples of designs against several different GPCRs, both class A and class B, and we have both antagonists and agonists. GPCRs are, of course, a very important class of drug targets, and we're excited about these new design methods as providing a new route to drug these important targets. We can also design proteins that bind to disordered protein targets. And here what we do is we allow the target to diffuse. It has a fixed sequence since we know what it is, while we're designing the binder. So in the darker color, you see the peptide target. It's disordered. It's sampling all kinds of different conformations, and we design a protein which basically wraps around this. You can see here. So we've used this approach to design binders to quite a few different disordered proteins and disordered peptides. These bind with nanomolar affinity in vitro, and we can show using localization experiments in cells that these binders bind to their targets inside cells. So disordered proteins play really important roles in biology. It's been hard to make tools that really specifically recognize them because they are, because they're disordered. It's hard to raise antibodies to disordered proteins because they get degraded. It's hard to make small molecules because there's not a cleft to bind to. But this the two-side diffusion approach is a very powerful way to solve this. One class of disordered or misfolded proteins are peptides that form amyloid that have been implicated in neurodegenerative disease, and we've designed proteins that bind to these peptides and can completely suppress aggregation. Here you see an example of a designed protein suppressing aggregation of amyloid beta, and here's another designed protein suppressing aggregation of tau. So we're excited about this as a sort of a general approach to targeting and then helping block the aggregation of amyloid-forming peptides. We've also been designing proteins to bind to small molecules, and this example shows you the design of binders which specifically bind very bright fluorescent dyes made by the Genelia group, and these are very widely used in imaging. Normally they have to be covalently coupled to the protein that you're trying to image, but now we have these proteins that bind them very tightly, so you just need to fuse the protein tag to your protein and you will label it. So this is an example of a 3-color labeling experiment where we have directed these 3 proteins to 3 different locations in the cell, and we can stain with all 3 colors simultaneously and very clearly resolve the different locations of these proteins. And so we're excited about these type of designs is really pushing forward what one can do in terms of microscopy-based localization and characterization of interactions in cells. One thing I should say is we can split these proteins into 2 parts, and then they only bind the dye when they come together. So we think this will be a very powerful way for measuring interactions between proteins. Currently, split proteins are used, but those often have some residual affinity for each other. So now switching gears from binding to the design of protein nanomaterials, my colleague Neil King developed methods for designing self-assembling nanoparticles. We've described many of these over the years. And then during the pandemic, he put on the receptor binding domain, which is the first approved fully de novo designed medicine, which we're very excited about. So to make larger particles, which is interesting for delivery and for materials generally, one has to break symmetry because if the largest regular polyhedron is the dodecahedron, which has 12 pentagonal faces. Now you're all familiar with from the soccer ball that you can expand the dodecahedron by inserting hexagons like this. But the challenge is that now the symmetry is broken, so the edge of a pentagon is different from the edge of a hexagon. So we need a design approach that can accommodate this asymmetry. And there are 2 approaches that we've explored. One is to build with subunits that have flexibility so they can adjust to encircle a pentagonal region or a hexagonal region as shown here. And this is a cryo-EM structure of such a design. This approach is called quasi-symmetry because the system is not totally symmetric because the subunits can have two different structures in two different environments. This consists of one protein chain, one unique protein, and 180 chains. Another approach is to put different protein subunits in different symmetrically non-equivalent position. So this is an example of this approach, which is called pseudosymmetry. Here we have 4 unique proteins and 240 chains. And these are very large particles. Here is AAV by comparison. You can see it really dwarfs them. These 2 new particles really dwarf it in size, which is exciting for delivery and other applications. What I've told you about so far is the design of soluble proteins. We can also design transmembrane proteins. And in particular, we can design beta barrel proteins that have pores of increasing sizes. And when these are inserted into membranes, as you might expect, the conductance through these pores grows considerably with the increasing size of the pore. Now, we can take small molecule binding proteins of the sort I've described to you already and put them at the entrance to the pore such that the pore closes down when the small molecule binding proteins are added. When the small molecules are added. And that's what you see here. This is the conductance through the pore without the small molecule, and this is with the small molecule, and the conductance is blocked. So we're very excited about this as a general way of making sensitive sensors for a wide variety of chemicals, because we can design these pores to have any type of interior we like. Now, since we're designing these from first principles, we can go beyond biology, which always has pores inserted into lipid membranes, and we can instead design pores that insert into silicon nitride chips into which holes have been drilled. So this is an example of a silicon nitride chip with a hole. This is an example of a protein that's been generated to fit into this hole. And this doesn't really look like any naturally occurring proteins because, of course, naturally occurring proteins never evolved to fit into holes in silicon nitride. Here you see the conductance of the chip only, and then when the design protein is added, it inserts and we get a stable current. So we're very excited about using these for a variety of applications now, including protein sequencing. So everything I've talked about so far involves design of proteins with single low energy states, but we can now design proteins that have multiple low energy states. So here, for starters, is a protein we call a hinge, which when it binds an effector, changes its state to this bent state. And you can see how if we build a shape like a triangle out of this protein, and then we add the effector, and it bends, we'll change the overall shape of the triangle to a square. And this is exactly what we observe here. Here's an electron micrograph of the original system. We add the effector, and this triangle changes into a square. We can do the same thing here now with the square turning into a pentagon. So this demonstrates we can make sort of these dramatic changes in architecture, so systems that will adopt 2 states that can be toggled by an external effector. Now we can use this to make medicines that can be turned off when desired. So here is a designed mimic of the IL-2 cytokine, which we've shown previously functions in animals, and that molecule has in fact been in clinical trials in humans. We can append to it a hinge, like the hinge I showed you before, which when the effector binds, clashes with one of the receptor subunits, pushing it off. And so this interaction can be turned off by adding the effector, because the way that the signaling works is that the molecule, the IL-2 mimic, brings these 2 receptors together. When the effector is added, it pushes one of the subunits away, which ends signaling. And so we can see this if we look at on the surface of cells when the original IL-2 mimic or the IL-2 mimic with the hinge is added, the two subunits come together very rapidly of the receptor. When the effector is added, they separate and diffuse freely. And we can quantify that here. This is the reduction co-localization upon adding the effector, and you see it happens very, very quickly. And this very rapidly shuts down cell signaling. So this is, if you add the IL-2 mimic here, and we get this increase in signal, but when we add the effector, it completely shuts off signaling. And we can use this to ask more detailed questions about what happens early on in IL-2 signaling versus later in IL-2 signaling. So there's also basic science implications of this. Now, we've been very interested in the last couple of years in designing new enzymes. And the basic idea is we start with a description of a hypothetical active site for this new enzyme that carries out a chemical reaction. And then we've developed methods, a method called RF diffusion 2, which basically builds a protein around this ideal active site, given only a description of the tips of the side chains that interact with the transition state and the transition state itself. And we've used this method to design enzymes that catalyze a number of chemical reactions, and the success rate is going up quite a bit. We generally, in all the examples I told you, we make multiple designs, multiple synthetic genes encoding these designs, and we make all the proteins, and we have to test all the proteins. But with these improved methods here, for example, we only had to test 96 designs to find one that had quite remarkably high kcat over km, much, much higher than had been previously observed for hydrolases of this sort. This one's interesting because it utilizes a metal ion, which is positioned between these histine residues here. So we now have so a lot of work in enzyme design is focused on sort of relatively activated substrates, but we've made a lot of exciting progress recently in designing enzymes that work on not so activated substrates. And one of the most interesting of these are de novo designed proteases that cut the peptide bond, which is a very stable bond, as you know, and it is very difficult to hydrolyze, and proteins and peptides by no means have activated bonds. And so here you can see we've made both zinc proteases that use a zinc ion, just as in the example I showed you. And here you see when we add zinc to a mixture of the protease with the substrate, we get complete conversion of the substrate to product. And here a cysteine protease has this. Here you see a rate versus substrate concentration of Michaelis-Menten curve, and we're getting the overall kcat over km is still relatively slow, so there's room for improvement. But we're seeing also, is not shown here, quite reasonable specificity for these enzymes. So we're very excited now about new classes of therapeutics that are not just binding proteins, but also incorporate a catalytic functionality like a protease activity. So you could destroy your target and your cancer cell oncogene or a virus, and thereby require much less drug than the stoichiometric amounts that would be needed to leave them, to just block them by binding. So we're also very interested in applications beyond medicine. So we've been very interested in trying to design simple photosynthetic systems for starters. So at the heart of photosynthesis are the photosystems, which have what's called a special pair of chlorophyll molecules in close opposition. And Nate Ennis designed a protein which holds these two chlorophyll molecules in exactly the same position. And now we can study the photophysics of the heart of photosystem without the huge complicated membrane protein. And we can design these into larger systems, such as these cubic structures with potential light harvesting ability. And we're now designing antenna complexes that absorb light at higher wavelengths that can then funnel their energy into the system, as well as catalytic groups that can take the utilize the oxidizing and reducing power from this simple photosystem to do chemistry. Finally, we've been very interested in designing proteins that form hybrid materials. So in nature, we have things like bone and tooth in our bodies that are made, and they're shells and seashells that are made by proteins interacting with inorganic compounds. And so we've been very interested, could we learn the rules for designing such compounds from scratch? And here is an example of a protein that's been designed to bind to be complementary to the hydroxyapatite lattice. And it has now been designed into this nanotube by Tracy Yu. And when we look at it by itself, we see here's the structure of the EM data on the design. And it forms this tube, and the tube is empty. When we incubate it with calcium phosphate, the precursor to hydroxyapatite, the tube fills in, and we get these tubes of hydroxyapatite in the middle, which are crystalline, as we can see from these images here. So we're very excited about now building completely new kinds of hybrid materials. And so we can extend beyond the sorts of minerals that have been used in nature to zinc oxide, for example, which is a semiconductor. And here you just see a designed assembly, which has a protein that mediates zinc oxide deposition on its surface here. And you can see in the presence of zinc oxide that this triangle fills in. So we're getting mineralization exactly where we want it. So we're excited about the possibility of this as a new way for growing semiconductors for perhaps for computing and other applications. So I've tried to give you today an overview of the applications of designed proteins that really now span a very wide range of areas, from medicine to technology to sustainability. And we are making all the methods we develop available. So I encourage all of you to explore designing proteins, or if you have things you might want to collaborate on, let me know. And this work was done by many brilliant people, so in my group and in the broader Rosetta community. So thank you very much for your attention.
Thank you, David Baker, for your wonderful talk. I'm Professor Mingyang Baek from Seoul National University, the moderator of this keynote session. So we are now moved on to the Q&A session. And to ensure a smooth Q&A session, we collected questions in advance during the registration process. And we have selected several representative questions, and we'll address them first during the session. So to help the camera captures the questioners, we kindly ask that you raise your hand and stand up when your name is called before asking your question. Thank you. So let's now connect with Professor David Baker, who is joining us on Zoom for this live Q&A session. Hello, David.
Hello, can you see me? I can hear you.
Okay.
Great to see you, Mingyang. Sorry I'm not there in person.
Great to see you. And thanks again for your wonderful talk. So as a moderator, I'd like to give a first question for warm-up. So from your perspective, to what extent do you think AI can contribute to protein design or therapeutic development? So if there are any areas that AI cannot currently address, and how do you think they should be approached?
Well, I think AI has really transformed protein design and protein structure prediction. So it's very clear that it is the appropriate approach for both of those problems. I think for drug discovery, AI is, I think moving forward, antibodies, will be designed on computers. Small molecules will be discovered using AI methods. I think some of the challenges now are at higher levels of biological organization. So, for example, predicting what effect a small molecule might have on a cell or on an animal, what the toxicity of a small molecule might be, immunogenicity. So I think AI is proving to be extremely powerful for structural biology problems, but of course, drug discovery is more than just structural biology. You can find a compound that binds tightly to a target, but it's still not a good drug. So I think those are the problems that AI is going to, those are kind of current research problems, I would say. Also, while progress is being made in designing, as I showed you, in designing antibodies against targets on computers, there's still work to be done there. And the design of small molecules to bind targets is still a very open research problem. So I don't want to give the impression that these problems are solved.
Okay, thanks for your answer. And we are move on to the questions submitted by Mr. Byung-Moo Lee, Executive Director at Yuan Corporation. So Mr. Lee, please go ahead with your question.
Okay, thank you for your great talk. I'm from Yuan Corporations. I have two questions about your presentations. Your research is openly shared on GitHub, which is very helpful. However, in today's open source ecosystems, particularly among startups, I'm sorry, and I'm sorry. Okay. I'm sorry. And so, particularly among startups, they really to release to only the training models without disclosing the training data, training method, or inference code. So I'd like to hear your thoughts.
Yeah. So that's a good point. So we release the code we develop open source. It takes us longer. The problem with training code is it's just often a lot harder to package. So for example, we just released the RF3 code. We'll be releasing the RF3 training code soon, but we're committed now to always releasing the training code in addition to the code, because we really want people to use our software and to expand on it. When we don't release the training code at the same time, it's not because we don't want to, it's because it's harder, because you have to package up all the data sets, so it's much harder it takes more work to make it pretty than to make the code itself pretty.
Yes, thank you. And the second question is about the regulations. I'd like to know your thought on what kind of regulations are needed to prevent ethical issues and misusing AI-driven research.
Yeah, I think So we've been thinking a lot about that problem, and we've been discussing with colleagues that actually the U.S. government asked us to hold a workshop to investigate that question. And I think the general conclusion was that right now, these methods have much more power to do good than to do bad, but that we should carefully consider when we're releasing new software what possible misuses would be. And so the community is very thinking about that problem. And the general consensus right now is that nature has evolved all these horrible viruses. I think it's going to be a long time before anyone can design a virus, if not ever, because it's harder than the drug development problem if you think about all the different steps a virus has to do. But there's a lot of opportunities for combating viruses with design proteins and design vaccines.
Okay, thank you.
Okay, thanks for your answer, David. And now we have a question from Mr. Changjun Bae from MFDS.
Thank you for your impressive presentation. I'm a regulatory working at MFDS here. I'd like to hear your insight on how regulatory agencies should prepare for the potential applications of medical products based on AI predictions during future health emergencies such as pandemic. Research is currently underway to proactively develop the vaccines and therapeutic antibodies by using AI technologies to predict the mutation pathway and potential risk of the future viral variants. In preparations for future public health emergencies such as pandemic, applications for the marketing authorization of predictive AI-based medical products may be submitted. We would greatly appreciate your insight on what preparations regulatory authority should be make in this regard.
Yeah, that's a really important question. The answer is that I think that, new therapies and vaccines that are generated using AI methods should not really be treated differently from new therapies and vaccines generated using conventional methods. One will have to, as a regulator, you know this better than I do, one has to balance the benefits from rapid approval with the risks. So as I said earlier, the AI methods are going to be very good at generating, say, antibodies that bind or small molecules that bind. But as far as predicting toxicity, those are still very hard problems. So I think the same type of safety testing will be required, at least at this point, for AI-generated therapies. So I think it will be important to go through the current procedures that you would use now with non-AI generated medicines. I think the difference is that the candidates will come up much, much faster. So if there is a new pandemic, very rapidly, we and others will be making new therapies and vaccines. And so you'll get them sooner, but then they will have to probably go through, safety testing in a similar way. And the time frame for that will probably depend on how large the crisis is that, if there is a new pandemic.
Okay, thank you. So now we will hear from Junhee Pyo from Kripia.
Thank you. I have two questions for you. You have actually revolutionized protein binder research area so far. So what is your research plan for next step?
Oh, yeah. Well, that's a good question. I mean, I talked about today some of the areas I'm most excited about. Things I didn't talk about are designing machines that can use a chemical fuel and carry out work. Designing new types of hybrid materials I'm very excited about. New catalysts, there's all kinds of chemistry I think that we can catalyze, including, as I mentioned, reactions that are important, like breaking down plastic, polystyrene and so forth. So there are a lot of good problems.
Thank you. And second one is actually beyond single chain prediction using like Rosetta Fold, proteins from multi-chain complexes within body. So in what direction do you expect with research field will develop in this area, and how could we tackle the challenges related?
Well, I think for predicting the structures of protein interactions, Minkyung actually has done really beautiful work in developing improved prediction methods. And, they are being used now to predict all the interactions in the human body. So I think we'll see increasingly accurate models of protein interactions and protein interaction networks, also proteins interacting with DNA. So I think we'll get an idea of all the molecular interactions in a cell.
Thank you.
Okay, thank you. So we have one more question from Mr. Jeongmin Choi, the Executive Director of Korea Biomedicine Industry Association.
Hello, thank you for your excellent presentation. So we understand you are working on protein-based drug discovery using generative AI, and there are some concerns have been raised about potential toxicity and immune reactions. How do you see the prospects for clinical success and scalability, and when might we expect some tangible results?
Yeah, that's a good question. Well, the biggest concern is immunogenicity. We have had some compounds in human clinical trials, and they weren't strongly immunogenic. For example, an IL-2 mimic called NEO2. But we're working now on design methods for greatly reducing the immunogenicity of design proteins by removing T-cell epitopes. I think we'll see an increasing number of design therapeutics go into clinical trials in the upcoming years, so we'll get a much better idea of it. But in general, we don't see strong immune responses from design proteins because they're very stable and very soluble. I think scaling is not hard. Making large amounts of design proteins is going to be probably easier than most biologics because they're very stable proteins and they're expressed at high levels in bacteria. I think immunogenicity is the biggest question.
Thank you.
Okay, thank you, David. So we have some time for live Q&A, I guess. So is there any question from the floor? Yeah, there's one question. Please introduce yourself before asking a question.
My name is Shaima Ahmadin. I am from Saudi Food and Drug Authority. I have two questions. First question about the safety and risk assessment. What risk mitigation strategy are applied to avoid of target binding across reactivity, especially in therapeutic protein designed like avoiding tighten cross reactivity in TCRs?
Let me just answer that question first. So we have actually been looking at the specificity of design proteins by mass spectrometry pull down in collaboration with Matthias Mann. And it's really interesting. Most of our design binders really only bind the target they were intended to bind. And then in far as MAGE and Titan, I showed you that we can design proteins that don't have that cross-reactivity problem. Now, they may have other cross-reactivity problems, so I think it will always be very important to do testing for that before going into the human body. But so far, things look really pretty good.
Okay. Second question, from the regulatory perspective. How should sponsor demonstrate comparability if the AI-designed algorithms evolve like in RF diffusion V1 versus V2?
Sorry, I didn't hear the first part of your question.
How should sponsor demonstrate comparability if the AI-designed algorithms evolve in the process?
How should the sponsor, what do you mean?
How to demonstrate the comparability? For example,
how to demonstrate that the models are better?
Yeah, for example, in RF diffusion of
V1
versus
so
V2.
You would have benchmark. Typically, with any of these AI models, you have a benchmark, and then you show improved performance on the benchmark, and that's how you demonstrate that a model is better. With design, you can't really know until you make things in the lab, but but it's generally you start with in silico benchmarks.
Okay. Thank you. So due to the time constraint, I think we have to wrap this Q&A session up here. So we apologize for not being able to answer all of your questions. And thanks, David, again for your wonderful talk, as always. And please give him a big round of applause to the professor, David Baker, for his valuable time. Thank you, David.
Well, thank you, Mikyung, and great to see you. Yeah, thanks, everybody. Yeah.
Thank you for inspirational keynote speech and engaging Q&A session. Also, we appreciate the moderator for leading this session smoothly. Please give another round of applause. Now we will take a 5-minute break, and the session 1 will begin at 11:35. So we kindly ask all participants to return to your seat, be seated by 11:35. Thank you. I will see you soon. may I have your attention, please? The session 1 of AIRIS 2025 will begin very soon. We kindly ask all participants to be seated. Thank you. Once again, may I have your attention, please? The session 1 of AIRIS 2025 will begin very soon. So all the guests of honor and participants in the foyer, would you please make your way into the hall and take your seat? Thank you. Once again, may I have your attention, please? The session 1 begin very soon. So all the guests of honor and participants, would you please make your way into the hall and take your seat? Also, please set your mobiles or other electronic devices to silent mode. The session will provide simultaneous interpretation service. Please note that channel 1 is for Korean and channel 2 is for English. Thank you, ladies and gentlemen. Welcome back. We will now move on to the session 1 titled Global Ecosystem for AI in Healthcare. Where do we stand and where are we headed? The session 1 will be conducted as a panel discussion. So I'd like to introduce our moderators first. Dr. Ki-dong Park of WHO and Jong-chul Ye, Professor of KAIST, will lead this session and lead all the panel discussion. So when the moderator introduces the panels, we kindly ask panels to come up to the stage and take your seat. So I'd like to invite our moderators, so Dr. Ki-dong Park and Professor Jong-chul Ye, would you please come up to the stage? Ladies and gentlemen, please welcome our moderators with a warm round of applause. So would you take the stage and invite the guests?
Yes, thank you for the master of ceremony. As introduced by our beautiful MCs, I'm Ki-dong Park. I'm from WHO based in the Philippines, Manila. As my title is Director of the Data, Strategy and Innovation. I'm trained medical doctor, trained as a generic public health physician, not specialized in regulator. However, I think I can entertain today's panel discussion for next 50 minutes. Professor Ye, why don't you introduce yourself?
Yeah, hello. I'm Jong-chul Ye from KAIST. I'm a professor at Graduate School of Artificial Intelligence, and it's really my great honor. In fact, this is my second time to moderate this panel discussion in AIRIS after the last year's 4 sessions. And in fact, over the 1 year, there are a lot of progresses in AI, especially in bio and health areas. So it's a really great honor, and also looking forward to hear a lot of opinion leaders' ideas about the vision in the healthcare AIs in the future.
Thank you. We have 5 panelists today. So I will introduce the first 3 panelists, and then Professor Ye introduce the next 2 panelists. First panelist is Dr. Talla Fakuri. She is the Vice President for AI and then Digital Policy at Parexel, focusing on real-world research. She previously served as Associate Director for Data Science and AI at the US FDA's Center for Drug Evaluation and Research, where she led agency's first draft guidance on AI in drug development and established the CDER AI Council. Earlier through her career, she worked as a US CDC as a Chief Statistician for National Health and Nutrition Examination Survey as an Epidemic Intelligence Service Officer. Second panelist is Mr. Bruce Church. He is a Chief Mathematics Officer and an EVP for Research and Early Developments at ITR. A founding team member of the both ITR and the GNS Healthcare, he is the principal inventor of the REFS platform and the leading expert in causal AI and then statistical physics. His career spans application from fusion energy to protein folding, and he has led major research grants, including $2.5 billion award from the US Department of Energy. Our third panelist is Mr. Hwa-young Lee. He is the Vice President and Head of the AI Business Transformation Unit at LG AI Research. He leads efforts to apply AI across LG affiliates and to identify growth opportunities in AI, biotechnology, and then clean technology. Since joining LG in 2004, as a software engineer, he has filled leadership roles in product management, business development, and strategy, guiding the company's adoption of the AI and the emerging technologies over 2 decades. Professor Ye?
Yeah, let me introduce the other two panelists. First, Mr. Pat Bayard. Pat Bayard is a senior regulatory specialist at Philips, focusing on AI in healthcare and software standard. He describes his work as a policy engineering, reaching the need of regulators and developers through standard, white papers, and training. With a background spanning software development, engineering management, and risk management, he is now co-chair AI committees at AAMI, ISO, CTA, and ADVAMed, contribute to IMDRF's AI group, and co-author the RIPS book on software as a medical devices. The final panelist is Mr. Simao Campos. He represents the International Telecommunications Unit, ITU, on the Secretariat of Global Initiative on AI for Health, a partnership of ITU, WHO, and WIPO, and serve as a counselors for technical standard and AI for health. He has worked at the ITU standardization sector since 2002, focusing on AI for health and operational excellences. He previously served as a counselor for Study Group 16, supporting the development of global standard in multimedia and digital health, and last Secretary of ITU WHO Focus Group on AI for Health from 2018 to 2023.
Thank you. As mentioned by Minister Oh Yu-kyung and then WHO Director General Dr. Tedros, AI, artificial intelligence, is rapidly reshaping our worlds. From healthcare to transportation, AI is rapidly moving from the laboratory to daily life. AI optimism is rising. In many countries, the AI products and services are considered as more beneficial than harmful. However, we should admit the concerns on potential harm of AI. To support its member states, the WHO, which I am working at, World Health Organization is in collaboration with ITU, International Telecommunication Union, established a joint focus group on AI for health in 2018. It has been migrated to a joint UN global initiative on AI for health in 2023, which is co-led by WHO, ITU, and WIPO, World Intellectual Property Organization. It aims to make AI in health safe, ethical, and inclusive from design to use. In 2023, WHO published its first guidance on regulatory consideration for AI in health. Since then, we have convened a working group on regulatory consideration to develop practical tools. Last year in 2024, we convened a strategic roundtable on AI in health during 77th World Health Assembly. It highlighted the need of the strengthening and harmonizing regulatory function of the AI-enabled medical products. The Ministry of Food and Drug Safety of the Republic of Korea, MFDS, launched AIRIS in 2024, which is a key platform to align regulation of the AI in medical products. WHO welcomes the initiative of the MFDS and then joined the inaugural session last year as a partner. This year, we are honored to co-host the second session of the AIRIS, AIRIS 2025, together with MFDS. We were empowered by the welcome message from Minister Oh, Prime Minister Kim, and then WHO DG Tedros during the opening session. Next 3 days, we will share latest trends in AI-enabled medical products. We will also discuss how to strengthen AI regulations through global collaboration. Before deep diving into many presentations, many heavy discussions, serious discussion, this session will take stock of our current position as a global community and examine the evolving global landscape of the AI governance in healthcare. Together with Professor Ye, co-moderator, and 5 panelists who have rich experience in regulatory space from different positions in the global AI ecosystem. I will entertain you for next. Initially said that host gave me 50 minutes. Now it's that they said you should cut down session in 38 minutes. So I'll entertain you for next 37 minutes to discuss this. Before asking questions to panelists, I would first invite Professor Ye for his opening comment as a co-moderator.
Yeah, thank you. So in fact, actually, after the last year, I remember I was doing the moderation with moderating the panel session. At that time, there was a FDA and the discussion with the MFDS in Korea, and about how to regulate this AI for healthcare product and etc. And after that, there are a lot of product discussions, and also MFDS has successfully launched new tools, regulatory tools to regulating the generative AI for healthcare product. I believe that's actually the first in the world. But anyway, so that was a very this IRIS platform has inspired a lot of those kinds of discussions about how to regulate it. So I believe today's session give another kinds of chance to, and with the WHO groups and world leaders, and how to impose and how to actually make these kinds of regulations in using AI for healthcare sectors. Yeah.
Thank you. We designed this session into 3 parts. First part, I will pose same question to all the panelists. The question is, what is the one AI regulation initiative you are leading now, and what impact has it had so far? Same question to all 5 panelists. Let me begin with Dr. Thalla.
Can you hear me okay? Yeah. Okay. Well, first, thank you very much for the invitation. It's great to be with everyone here again. When I joined last year, I was with the US FDA and we helped co-organize the conference, so I feel very happy to be here for the second time. Some of the initiatives that we're seeing around AI for drug development specifically have recently come from the US FDA, but also from EMA. So earlier this year, FDA issued 2 draft guidance documents, one on AI use in drug and biological products development, and the other was specific to medical devices. The agency, I was with FDA until the end of June, had received many comments from the public on both guidance documents, and I know that former colleagues are working to update these guidances. Importantly, and I think for the purposes of this meeting, FDA meets with other regulators on regular cadence, specifically with EMA colleagues. And these meetings are essential to ensure that any harmonization that happens across the globe are consistent, right? And are based on the experiences of the regulators with submissions that they've received containing AI components. So that was a lot of the work that I led previously when I was with the FDA. Currently in my new position with Parexel, we provide basically regulatory consulting to different organizations on their uses of AI, the responsible use of AI to generate data that is considered credible and could be trusted by the regulators, and happy to elaborate on that.
Is this on?
Yep.
Yeah. Well, thank you to the organizers for inviting me back. I was here at AIRIS 24. My world is a little bit farther away from the regulatory space in that we're doing the discovery of biology using AI. It does touch on some of the same issues in terms of fairness and inclusion and bringing as much data from people and trust of that data. And so those are places where the conversations that are happening here can have a big impact on our world and ability to collect the multi-omic data across many different patients, across people from many different parts of the world. It's that variation that we learn from, and so it's very important to have that trust. But on the regulatory side, one change that has happened in terms of downweighting the importance of animals in testing is a place where digital twins can also play a role in reducing the amount of animals that are used. We're currently exploring using this for placebo arms and of experimental regimes in animals and other model systems. And I think this will, in the long run, dovetail very nicely into the observational data work, the digital twins that we're building for humans. Thank you.
Hello. As an employee in the AI company, so it is useless to say how much the data is so much important to train an AI model. There are a lot of open data set out there, not only just bio and healthcare sector, in large language model, large vision model, there are a lot of open data sets out there. And a lot of AI scientists and data scientists, they just release this data is available for everybody, it's open source. But in reality is, when you look into the data set and the copyright, the documents, it is not allowed to use the commercialized version of the AI model, or sometimes it is not allowed to use for specific purposes. So in this case, in this kind of circumstance, we cannot trust the Hugging Face based on the open source data. So in this case, it is really hard problem, because in one data set, there is another nested data set, the depth level is more than 78 depths. So in that case, one of the data set that the copyright is not allowed to train the AI model. You cannot use that AI model for the commercial purpose. But there are so many the mistakes when some of data scientists and AI scientists release the data. So in your industry, it is almost impossible to figure out what kind of data is commercially and viably to train the AI model to break a new scientific breakthrough in the bio and the healthcare sector. So these type of the problem I would like to touch. So the legal attorney cannot solve all this problem because there are more than 10,000 and 100,000 data sets out there. So the AI model, we usually call the legal agent, the AI model, we're gonna figure out, wow, this data is available for everybody. This data is limited for this purpose only. This type of the ecosystem and the system is required to train the right and the ethically and safely train a model for the bio and healthcare sector.
Is this working? All right. Usually when I start talking about AI, the technology knows that I'm talking about it, and so, won't let me log on. It makes sure my microphone isn't working, something like that. So the technology is giving us a pass, at least for this session. So, but watch out. For those of you that don't know me, I like trying to use humor when I'm presenting. You just saw an example. I am not claiming to actually be funny. I'm just saying that I will try to be funny for some of these topics. One bit of trivia that I did want to, not related to AI, but I wanted this audience to know, is I had noticed yesterday was the 78th anniversary of the very first computer bug. I saw that notice last week, and so I thought that was just interesting as I'm traveling here. The very first computer bug was 78 years ago, back in 1957. And at that time, the computers, they used relays. And for those of you that don't know, a bug is really meant to be the moth got caught in the relay. And so that was a bug in the operation of the computer. Clearly, the software people were, abusing that. But I just thought it was a great irony of right before we start talking about artificial intelligence and very complex computer systems, it's the anniversary of a moth, the anniversary of a bug. As I'd mentioned, I like to use humor in my presentation. I forgot to to point out to you, take a look at the spelling of my last name, okay? The universe tells me that I should be AI R&D. It's burned into my name. Clearly, I'm not good enough to be a professional comedian, and that's why I write standards, because this stuff isn't very funny. Have lots of other things that I'm hoping to take and talk about over in the next couple days. I do a lot of work with standards, but as a former software engineer, I really like reusing, what's already been written. What we did for software, we can also take and do for standards and try to get this to happen much, much faster. In the example that I like to use is, hey, when we're talking about getting a sample, getting some information and feeding it into our system, we want to make sure that it's good. And so I propose that we handle data according to these rules. We should keep records and retain information on the origin of that sample. The sample sourcing, any processing that you need to do, preservation so it doesn't go bad, any testing or other handling you need to do should be done in a safe manner and protected against contamination. Does that sound like good data techniques to start with? Okay, yeah. So that's from a standard I found on, actually an IMDRF guidance document on taking care of biological tissue samples. My point being is for a lot of this stuff, we know what good looks like. The challenge is translating that good and seeing how that works out for software and works out for data. And I'll stop with my soapbox for now.
Well, good morning to you all. I'm very glad to be here, Simon Campos from ITU. Thank you for the organizers to inviting me to be here, a part of this journey with WHO and now starting with WIPO, with the Global Initiative. ITU is a sister organization to WHO, so WHO is the UN Specialized Agency for Health, ITU is for information communication technologies, so this is more or less the scope of the work. We are a facilitating platform in a way that we try to help members across the world to come together and facilitate discussions, find solutions. My work area is directly with technical standards, so I am really very well aligned with Pat here. I don't use humor so much, I try, but take Pat very seriously. Huh? I know him for a while, and he's quite serious in his humorous ways. Anyway, the work of ITU with WHO in the focus group that Kidong mentioned just before was to try to bring thoughts around the benchmarking of AI solutions in health. We started with a technical mindset to very quickly realize that it is not only technical, there is other regulatory, societal, and aspects that comes with AI and health. So it was a little bit of a shock and a very great learning opportunity that you had with that focus group initiative to create a community across all different parts. So we had the regulators part of the group, we had health practitioners, we had data science scientists and people from the standards communities trying to work together and develop a framework. It's not a final framework, but it was a pioneering one, I think, in trying to bring all this together. Pat mentioned some of the concepts being already existing in older areas and trying to translate. And the translation was a good part of the effort in trying to understanding ourselves and bringing the concepts together, understanding the different ethical, regulatory, the technical aspects, and also bringing that into practice to make sure that whatever we bring fit into health use cases and bring meaningful results and can be used. So this was part of the venture that we had with the focus group that we are translating now, bringing WIPO. We focused more on the, let's say, benchmarking aspect. Now try to enlarge that to go towards the wider adoption and implementation within countries. I'll pause here and thank you.
Thank you, 5 panelists. It is confirmed that we have very good mix of the expertise from different part of the AI regulatory ecosystem, from regulator, R&D side, data, and international organization. With that, let's move to part 2 of the panel discussions. So we prepared the question specific to each panelist. So let me begin with the question to Dr. Talla. The question for you is what lessons from drug development can guide global rules for AI in health? How should regulators and developers work together to speed safe approvals?
Yeah, so AI use in drug development is so wide and very ubiquitous. It starts from areas that the generally regulators don't look at, like discovery, and then it moves into areas that regulators are very interested in, non-clinical, clinical, post-market, and advanced manufacturing. Because the uses are so ubiquitous and because AI is literally a tool, it's very hard for regulators to regulate that specific tool. So the context of use is very, very important. When I was with the FDA and when I used to have conversations with other regulators, the emphasis was always on why are you using this specific technology? How are you using it? And then approaching it from a risk-based framework where the emphasis is on what is the risk that's associated with AI use for that specific context of use. I'll just give an example from the FDA draft guidance. a sponsor might be interested in using AI to identify patients that can be monitored after dosing in their home versus be sent to a hospital for monitoring, right? This is potentially a high-risk use case, especially if the sponsor will be relying solely on the output from the AI model. AI can also be used to recruit patients into clinical trials. AI can be used to identify specific biomarkers. AI could be used for multimodal types of uses of AI. It could be used to generate external control arms or digital twins. The considerations for each of these uses are really different. So you can't be rigid. You can't have over-regulation that would prevent the use of AI in many low-risk uses that are low-hanging fruits that can increase efficiencies of the entire process and help accelerate the development of safe and effective drugs. So I think the lesson that I've learned over the past few years is that regulators should really take a risk-based, proportionate approach to AI use that is anchored in the specific context of use. It can't be something that is too broad where over-regulation can prevent the useful use of that technology.
Okay, so let me ask a second question to Mr. Bruce George. I think it may be related to the question to the first one. So, yeah, especially in drug approval, the evidence-based medicine is a very important keyword. So you have been working in R&D sector for many, many years, and you are a chief mathematical officers and also in charge of R&D sectors. So my question is, from the R&D perspective, so similar to the drug approval process, what do you think is the top 3 pieces of evidence should the regulator require to judge that this AI product is safe and reliable?
Well, again, I just like Tala said, it depends on where you're looking. So for example, Dr. Baker talked about designing new proteins that do interventions. Those can be tested by the safety and efficacy can be tested by the way we've always done that. And in my world, where we're doing very complementary work, where we're trying to identify what the impact of that intervention on the biological system is with a digital twin, there again, it can be tested by doing experiments, just as we always do. Where it gets more interesting is when the AI is the intervention itself. So for example, by risk stratification, decision AI tools to drive decision-making around patient care, that's where the regulatory piece gets a lot more interesting, at least from a logical or a mathematical perspective. I look at it from that perspective, because that's my point of view. And I think that's where causal AI can play a role. It can help us mathematize or write down in a logical framework what we mean when we say an algorithm is fair. What do we mean when we say that it's safe? These are interventions that we are doing, and so causal AI is really about taking the typically observational data that we have, the data that's easy to generate relative to experimental data, and trying to pull as much information about what will happen when we intervene, when we drive a decision with that. And so, for example, the ability to have the math to answer counterfactual questions where I say, what would have happened to this patient if they had gone into the other arm? What would have happened? We take all of the other external sources of noise that drive different outcomes, and we hold those fixed, and we ask what would have happened. That's a place where a causal framework can help you tease out what's fair and what's safe. So I think there's an opportunity to weave that causal perspective into the regulatory framework.
Next, I will ask to Mr. Hwa-Young Lee. Since you are leading AI business in this LG big company, so what role should industry play in building international cooperation for an inclusive AI for health ecosystem?
Yes, I think industry, especially the AI company, need to build an open and transparent ecosystem rather than just building and delivering the state-of-the-art AI model. Especially the AI company is more rigorous about performing like this. It is good to introduce about the LG's vision of AI first, to explain about why this is so much important. We would like to build an artificial expert intelligence rather than the artificial general intelligence, which have enough level of intelligence in each domain that can actually help the human domain experts. So while collaborating between the AI model and the domain experts, more and more data are going to be generated. And those data are going to be used to train the AI model again. And the AI model is going to smarter again. And the AI model is going to help the domain experts more and more efficient and effective way. So in effective way. And this kind of the virtual cycle is really important for the long-term sustainable the growth and advancement of the domain expert and the artificial expert intelligence, we think. To accomplish this goal, the openness and the transparency are the key of the key factors, I think. LG has been offered our own AI model to the public, named XR1 Path, which is designed to stratify patients during the clinical trials. It is free to use and distributed via the Monai platform by the NVIDIA. Through releasing through releasing the technical report, we unveiled what kind of data we used, how we could train the model, and how much better performance it has under which circumstance. So everything is open and transparent. Now the number of the download is more than 10,000. So meaning that a lot of domain experts trust that AI model. So we truly believe that we move one step further to build the virtuous cycle in the clinical trial domains. So I think that the role of industry, as I mentioned in the vision of the LGA research, is not just building a state-of-art the AI model, but also thinking of how to build an ecosystem that could grow together in terms of the AI and the human domain experts. Thank you.
So let me ask a question to Mr. Pat Bayard. So you have been working on standardization for many years and also quite interested in AI standardization. My question is, nowadays there are so many developments in AI in every day. So in considering those kinds of fast development, what do you think we need to do to standardize? And is it possible to standardize AI, especially for the healthcare sectors?
Well, I actually, for the next session, have a 20-minute presentation on that question. And mainly just so that you have slides to refer to, when someone asks you a question 2 months from now, you can go take a look at the slides and say, oh yeah, hey, look, there's a standard working on that. So I'm going to sort of answer this question in a different way. And just talk about how some of the ways that I have been leveraging the standards. I'm one of the industry members on the IMDRF AI committee, and IMDRF is currently working on developing a lifecycle document. And so I volunteered for them, hey, instead of creating a completely new lifecycle document, I have 6 different standards that have already been published with different life cycles. Maybe we should take a look at them. I'm not saying any one is perfect for us, but instead of trying to invent something new, again, the software engineer in me says, let's take a look at previous products and see what we need to take and build. In a similar way, the FDA, Health Canada, and MHRA, you might remember they've published a paper on basically some guiding principles. And well, I sat down with those guiding principles and I made a little spreadsheet that maps each principle to what standard can you find some of the text for that. Again, I'm not saying adopt the standard and you're done. I'm saying, hey, these are some initial drafts we can take a look at and review collectively, and then decide what we want to take and move on or not. Let's see, there was one more. Yes, I also wanted to mention, and some of this is harvested from standards and some isn't, just personal experience. But what we noticed, you might remember that of course, we have to do adverse event reportings when we find out something bad has happened that might have involved one of our devices. And IMDRF has a list of adverse event codes. Well, a couple of us thought, ooh, it's an interesting idea. For artificial intelligence, it can fail in different ways. You know, given some of the standards work that I'm doing on risk management, yes, it can fail in different ways. And so, ooh, do we need any more details in the adverse event report? You know, and having some categories in there for the kind of failure for that AI. So got together with a couple other companies. We're doing the final polishing on it, and then we'll be submitting that to IMDRF for them to take and consider. And again, it's not us inventing something new. It's taking what's already out there, reusing that, customizing it for our kind of application. So, and if you have questions for me after this, I know that we're running low on time. There's enough hallway conversations and lunches to talk about this some more. Thank you.
Thank you. The next question is my colleagues, UNA sister agencies, CMO. So since 2018, WHO and then ITU has been collaborating in this area. And then ITU is you are covering, convening summit every year, AI for Good. So what key message came from the AI for Good summit, either this year or the last couple of years? How should they guide international standards that link regulators, industry, developers, and health systems?
Thank you. Yes, as we mentioned, we started this journey in 2018 with the focus group focusing on benchmarking. Why benchmarking? Is because people were talking about AI in health, but we didn't see bringing them to the market. So we started thinking what could help, in particular regulators, how to bring things into practical use. We spent these 5 years focusing on, well, developed the 36 deliverables from that work, and they touched the ethical aspects, they touched the regulatory guidance, they touched the technical aspects of use of AI, as well as the deployment and health use cases there. But we had more of a focus on actually the developer side, so people that would be trying to bring this to the market, what kind of a guidance could give them. For example, on the regulatory side, WHO led the development of these guidelines that were published by WHO, but also as focus group on the regulatory guidance that had checklists about what people should look into in order to facilitate when they try to clear their products, things that they should look up front. In addition to that particular deliverable on the regulatory aspects, we had 2 other documents. One, for example, mapping the essential IMDRF principles into the use and in practice, as well as checklists for helping with auditing of implementations. So these were examples focusing more on the developer side. We came to the conclusion that even though we have a rich set of materials, let's say a pioneer framework developed there, we needed to further the enabling elements of this guidance to also cover the regulators and the users and in countries in terms of the more, let's say, institutional users, not only the developers. So we wanted to invest in that direction. As well as for the other types of guidance to, in particular, the use cases, how this can be realized and how to scale up and implement. So, in the meeting that we had this July, I think we had a workshop on one of the days, and some of the key highlights, I would say, a call for these more, let's say, templates to be developed, for example, for the regulatory framework, model laws that would facilitate the country. So to give best practices and example on how people could, let's say, shortcut the development time and considerations for not only how to, let's say, put products into the market, but also the data access frameworks to open data, the intellectual property aspects, what could be the different approaches. There is a lot of, I think, demand asks in the market for people, how should we do this, So model regulatory frameworks that could be helping on the IPR side as well. I think Mr. Lee mentioned some of the difficulties. He's not the only one facing those difficulties. A lot of people are having a lack of clarity on how to approach this and what could be, if you could have some sort of interoperable approach to the regulatory frameworks that would facilitate greatly the greater deployment of safe AI in health. So I think these are some of the things that came up from the discussions we had at the workshop in July. Also, there was a call for more investment in open science. I think this was a requirement that, or something that would greatly help further the availability of AI for, in countries and fostering this across the different jurisdictions. Thank you.
Thank you. We have less than 1 minute. But I think we can extend 1 or 2 minutes for closing. So this session is kind of the induction session. After lunch, you will hear some more presentation on the detailed example, detailed practice, and the real-life discussion. And then tomorrow is how we will discuss how to strengthen collaboration. I'm hearing repeatedly the importance of the data governance and then the interoperability, and also as a regulator should take risk-based proportional approach and then move away from over-regulation part. And then if we can agree on this, or if we can share some examples of the model law or some model regulatory framework that will expedite our collaboration. Once regulator approved, AI enables medical device, AI enables something, public health sites will scale up and they will address equity issues and then healthcare quality issues. So I think this is a good start of the 3 days workshop. But I should invite Professor Ye for his summary and the closing.
Yeah, so regulation is very difficult. As I can see, we need more time for further discussions, I believe. But however, even though this is a difficult topic, and I think the common message from today's discussion, I can see there are 3 or 4 keywords from the common discussions. The AI and regulation should be open. Openness is important. And also those regulations should be based on some evidence. This is similar to the drug approval process. And also we need a standardization or some kinds of benchmark to make this kind of thing transparent. I believe this 3-day workshop, a 3-day conference will give a good opportunity for further discussions. Yeah, thank you very much for all the panelists and for all the audience.
We appreciate all the panelists and the moderators for making this session more meaningful, enriching, and rewarding. Ladies and gentlemen, please give another round of applause. Thank you. You may return to your seat. Thank you for thought-provoking messages and addresses. In the afternoon, there will be even more enriching and rewarding experiences and sessions, so we hope to see you in the afternoon. So before closing, I'd like to give you the announcement of a luncheon. The luncheon for invited guests will take place in Inspire Ballroom B, which is located in the next room. And those invited have been notified in advance, so participation can be confirmed in the yellow sticker on your name badge. For VIP guests seated at the table 1 to 5, we prepared the luncheon in the Valley Room where you had a tea time in the morning. So session 2 will start at 1:40 p.m. in the afternoon, so we kindly ask all participants to return to your seat and be seated by 1:40 p.m. So please enjoy the meal and have a great time. I will see you in the afternoon. Thank you.
사운드만 조금
begin in a few minutes. So all the participants and distinguished guests, please make your way into the hall and take your seat. Also, please set your mobiles or other electronic devices to silent mode. This session will provide simultaneous interpretation service. Please note that channel 1 is for Korean and channel 2 is for English. Once again, may I have your attention, please? The session 2 of AIRIS 2025 will begin very soon, so we kindly ask all participants to be seated. And please set your mobiles or other electronic devices to silent mode. Thank you. Once again, may I have your attention, please? The session 2 will start very soon, so all the participants in the foyer, would you please make your way into the hall and take your seat? Ladies and gentlemen, all participants, would you please make your way into the hall and take your seat as we proceed with the session 2 right away. Thank you. Welcome back, everyone. I hope you enjoy your lunch. We'll now begin the session 2, titled Latest Trends in AI-Enabled Medical Products, focusing on promising R&D technologies. The session 2 will be chaired by 2 moderators. So Professor Jongcheol Ye from KAIST and Ms. Mongji Chung from WHO will lead this session. So I'd like to invite our moderator to the stage. Would you please come up to the stage? Ladies and gentlemen, please welcome them with a big round of applause.
Okay, good afternoon. So, welcome to session 2, latest trend in AI-enabled medical product, focusing on promising R&D technologies. This session will highlight recent innovation in AI applications across drug and medical device development. I'm Jongcheol Ye from KAIST. I already introduced myself, so my co-moderator, please introduce yourself. Yeah.
Thank you, Professor Ye. Good afternoon, everyone. Hope you're not in food coma yet. This is Manji. I'm the team lead for research, ethics, and innovation in WHO Region Office for the Western Pacific. There are 38 countries and territories within our jurisdiction, and we serve them in strengthening research and innovation ecosystem. Over to you.
Okay. So this session, we have 7 invited speakers for the speeches. And in the first part of this session, there's 4 speakers will speak, and then there will be a coffee break, and then 3 speakers will follow. So let me introduce our first speaker, Tala Fakhouri. She's a Vice President Consulting AAD Policy Real World Research par excellence. She's going to talk about latest AI technologies and regulatory trends in pharmaceutical field. Yeah, please.
Hello, everyone. I walk a lot, so I'm happy I have a handheld, but hopefully I don't accidentally throw it. So I'll be mainly focusing on FDA regulations as it pertains to AI, but before we get into the exciting stuff of regulation, I just wanted to take a step back and kind of talk about why I think we're all here. This is Moore's Law. Many of you may be familiar with Moore's Law. Moore's Law tells us that the number of transistors in a computer chip basically doubles approximately every couple of years. AI has actually surpassed that. Moore's Law is a proxy for innovation. So from the 1950s to the 2010s, you can see that there was this exponential increase in innovation across the board. In the meantime, and over the same time period, this is what we, and when I say we, I mean the entire drug development ecosystem, this is what we've been doing. We've been doing Eroom's Law, which is literally Moore spelled backwards. EROM's law tells us that the number of new molecular entities per $1 billion that's been approved by the FDA has actually declined precipitously over the same time period. So as societies are becoming more innovative, the actual process of developing a drug has actually become less efficient. There's been a lot of excitement that perhaps AI can help flip this trend, that maybe AI is the tool that will help us become more efficient in developing safe and effective medicines. But is it hype? So I want to consult with my friend, the Gartner Hype Cycle, to talk about what is hype or what's not hype. You may have also seen this Gartner Hype Cycle. What Gartner tells us is that every innovation goes through 5 phases. So you could think about a specific innovation trigger, think about the Human Genome Project, or CRISPR technology, or gene therapy, or how real-world evidence will substitute the need for randomized clinical trials. This is when something new happens, we all get really excited, and then over time, our excitement takes us to that peak of inflated expectations where we realize that things are actually harder than what they seem. Some things will actually fail, not everything will work. And so a lot of people will go through this trough of disillusionment. This doesn't work. Why am I using this? But this is where most people and organizations want to quit. This is when you should not quit, because this is when you can kind of calibrate, figure out what works, what doesn't work for what you're trying to do, and how you could optimize it for your specific work process so that you can get into that slope of enlightenment and eventually the plateau of productivity. How does this apply to AI? When I say AI, I think most people are thinking of generative AI. And you're probably thinking about these really messed up images with hands that have 10 or 20 fingers. And you're thinking, yeah, it doesn't work. We're probably in that trough of disillusionment. But AI is not just generative AI. AI is many things. Many of these tools have actually been deployed in drug development for decades. So if you look at deep learning, as we heard this morning from the keynote speaker, has been integrated into the work that we do across the entire drug development life cycle for decades. Literally since the 80s and 90s. I used to code and I would do AI before I knew that it was AI. It was just machine learning. So a lot of us, I think, are in the same boat. And then if you look at new things like causal AI, I would argue, some might disagree, that this is in the innovation trigger. We're all excited about causal AI. AI that would actually help us figure out cause-effect versus just correlations. And then generative AI is going down that slope. I would say probably it's in that trough of disillusionment, and we're trying to figure out how to optimize it for the work that we're doing. Okay, so AI is many things. It's not just generative AI. It's been around for a long time. It's been deployed across every single stage of drug development. This is borrowed from an FPIA position paper where you can see that they list where AI is used all the way from drug discovery, non-clinical, clinical research, in regulatory submissions and approval, and the 2 bars on the bottom in R&D and CMC. AI is used across every single stage of drug development and has been integrated for many years. So this whole thing of AI exceptionalism, that's actually the hype, right? Now, FDA has seen AI being used across all stages of drug development. This is as of June of this year, so I was with FDA until the end of June, and these were the numbers that were presented by the agency at this time. So FDA's CDER, which is the Center for Drugs, has received over 800 submissions with AI components from 2016 until 2024. And if you look at the table, so you have the years 2016 to 2024, and then The totals are on the bottom. You can see that there was only one submission in 2016 and over 300 submissions in 2024. This tells us that industry has been really excited about using the technology, and they're integrating it into the work that they're doing, and they're using it in very different ways. So on the bottom are example use cases of AI. AI is used to select patients that may benefit from the drug. AI is used for outcome prediction, for pharmacometric modeling, digital twins, PV, and what I'm not showing also in advanced manufacturing. The graph on this side is by therapeutic area. Most AI uses that were submitted to the FDA were in oncology, which is not surprising. Okay, so this again tells us that AI is not all that, it's not hype. It's been used for many years, and this is starting to translate into what regulatory bodies are seeing. But this is all happening with this backdrop of a really complex regulatory landscape. This is a slide that's not specific to drug development or medical product development, but this just shows the number of AI-related bills that were passed into law by different countries across the globe from 2016 until 2024. There's not a lot of harmonization. There are so many different regulations across different countries. And if you're a drug developer or a medical device developer, you have to do your work against this complicated background, right? And this is just for AI. AI needs data. AI is a data monster. It needs a lot of data to produce outputs and to train the models. Data protection and privacy legislation is also complicated, right? So this shows us legislation, and the most extreme is no legislation as it relates to data protection and privacy across the globe. And again, you can see that there are a lot of legislations. They differ across different regions, different countries. You could think about HIPAA in the United States versus GDPR in Europe, and how these two are very, very different, and the impacts of these different types of data privacy legislation on the availability of data to train good models. So this is all to say the landscape, the regulatory landscape for AI is complicated. So against this landscape, what has the FDA been doing? The numbers that I showed you with 800 submissions received by CEDAR, these actually came from a paper that was published in 2023 by my former colleague, Dr. Chi Lee, in the Office of Clinical Pharmacology. So FDA is tracking the number of submissions, and I know that they're planning to update this paper. That was in 2023, but FDA's work around the regulation of AI in drug development actually began in 2019. FDA's CEDAR, the Center for Drugs, established an AI steering committee, a working group to develop policies on AI, and then began tracking AI use cases. All of that work was also informed by many expert workshops that were conducted with Duke Margolis, through the CERCES that the FDA has, and then there was a workshop last year in August. I think you'll all have access to these slides. If you click on any of these icons, it will take you to the summary of that specific workshop or meeting or paper. So FDA, based on these expert workshops, published 2 discussion papers, also in 2023, one that looked at AI uses across all stages of drug development, and another was very specific to advanced manufacturing. The discussion papers ended with a list of questions to sponsors and industry, asking them about their experience with deploying AI tools and asking questions to provide FDA with information on where regulatory clarity will be needed. Then in 2024, March of last year, all the medical product centers at the FDA published a white paper that looked at 4 areas of focus for the FDA over the next few years as it pertains to AI use in the development of medical products or when AI is the medical product. Again, you'll have the slides, you click on any of these things and it will direct you to that publication. Then late last year, FDA formed a CEDAR AI Council to manage the internal use of AI amongst FDA staff. So if you've heard on the news about ELSA came from something called CEDAR GPT. CEDAR GPT was managed and it's the processes where it was used by the CEDAR AI Council. And then the CEDAR AI Council also looked at policy development as it pertains to how sponsors are using AI. And then finally, the AI draft guidance that was published in January of this year, it was 2 guidances came out on the same day, one for drugs, which is shown here, and another one was for medical devices. So this is all to say that FDA's CEDAR has been working on AI policy since 2019. The effort has been ongoing for many years. So when the draft guidance came out, this didn't just emerge from thin air. This was informed by several years of engagement with industry. Okay, so I want to focus the next couple of slides just on the draft guidance. The draft guidance provides a risk-based framework for how to establish and evaluate the credibility of an AI model for a specific context of use. Credibility, just very briefly, is trust. How can regulators trust the output from a model? This is what credibility means. Context of use means the role and scope of AI use for a specific context of use. The guidance was partially based on, as I'll show here, partially based on the engagements with industry, also reviewer experience. So FDA reviewers have been receiving AI submissions since 2016, and then by existing frameworks. So Pat, you're gonna like that. The guidance relied partly on the ASME V&V 40 for a part of the communication around assessments of model risk, AI model risk. This is because, like Pat said this morning, there are so many standards and many frameworks. Our scientific thinking literally applies to AI in any type of modeling technique. Some things are unique and different, but for the most part, the tools that we already have to evaluate credibility of model outputs still applies in the context of AI. And most AI uses that we're seeing in drug development are machine learning models. They're not generative AI, at least not yet. Okay, so that guidance goes through 7 steps. I'm not gonna go through them in detail, but I'm just gonna highlight a few things. It starts with literally the most basic step. What is the question that you're trying to answer with AI? When I was at the FDA and I would do consults for regulatory submissions, it was really surprising how many sponsors would come in without saying what they're using AI. It would just be like, we used AI. But what are you exactly doing with this model? Also, what is the context? What is the scope? Will there be a human in the loop? Is there any other source of data, source of evidence beside the evidence from AI that the regulator can also look at, right? So the first thing is, what is the question that you're trying to answer? What is your context of use for your AI model? And then from the VV40, I'll show that on the next slide, is the assessment of AI model risk. This step is very important because what FDA reviewers do is they rely on their and your assessment, the developers, of model risk to make a plan on how they will establish the credibility or trust in the output from that model. And then what FDA will ask for will depend on model risk. So if you're using AI for a very high risk use case, context of use, Regulators will ask you for more information, more documentation, potentially more data. They might want to see your model, not always. But if it's a low-risk use case, most people at the FDA will say, thank you, I don't need to see anything. You could keep the records in case you're inspected, then the inspectors might ask for more information. So step 4 is developing a plan to assess the credibility. Then here there's a pause in the guidance where it says after the sponsor develops a plan, they should come talk to the agency. There's a lot of emphasis on engagement with the FDA. This is the model risk gradient that is presented in the guidance, which again is derived from a standard that was developed for computational models and medical devices called the ASME V&V40. On the horizontal line is model influence. So that's how much am I relying on the output from AI to make a regulatory decision? Am I only relying on AI or partly relying on AI? And the guidance walks you through 2 examples. Then before I show you that, the vertical line is decision consequence. What happens if I'm wrong in answering the question of interest with all of the data that I have generated from AI or otherwise? So you can think of examples where I might be relying solely on AI, and if I'm wrong, a patient dies. That's the extreme. Then model risk is really high. If model influence is low, the other extreme, I'm not really relying on AI that much, but a little bit, and no one's going to die if the model is wrong, then your model risk is low. So this is a gradient, and based on that gradient, the level of oversight, the stringency of assessments, and so on will differ, which means FDA will ask for more or less based on model risk. The guidance was published in January. FDA received a lot of feedback. I think it was, I have maybe a slide on that, over 1,400 comments on that guidance. The comments are available to the public. You could read them. I read all of them. There was a lot of enthusiasm for the guidance. Folks liked that it was risk-based, that it was high-level, that it provided flexibility. Of course, a lot of people wanted more detail, more use cases. And this is where I always encourage my industry colleagues to publish your use cases on how you establish the credibility of your models. Publish them in the literature, share this information, because this is a new space where we're all learning. The guidance, of course, as you know about guidances, they're not, it's just recommendations by the agency. They don't add new requirements, so there's nothing new that anyone has to do. It just works with the existing regulations and requirements that the FDA has. And the guidance also emphasizes the importance of early engagement. In fact, it ends with a series of different engagement pathways that are available to sponsors, again, anchored in that context of use. If you're using it for PK/PD modeling, talk to the MIDD program. If you're using it in advanced manufacturing, talk to ETP programs. So it provides a list of who to engage with and their contact information. Right now, I have 10 seconds. I don't think I can do that in 10 seconds, but the agency is analyzing the comments and updating the guidance so that it can move from draft to final. The agency is also, and this is all public, been announced, the agency is writing guiding principles for good machine learning practice that are based on the IMDRF guiding principles. Then, if you've seen, FDA has a digital health and AI glossary, which is critical for harmonization across all medical products, but also with other regulators. And then there's a workshop happening later this year in October. Anyone can sign up, and you could watch it virtually if you can't make it in person. A final slide reminder. AI can truly be transformative if we can scale it in all the right ways. I don't have time to talk about that today, but from my experience at the FDA, both FDA and EMA are largely aligned. People would usually say there are differences. There aren't really that many differences between the regulators, which is a great first step towards eventual harmonization. Some things will complicate the work that regulators are doing, like GDPR in Europe. And then another area where there might be differences is the level of data and documentation that regulators may ask for. And that's all I have. Thank you.
Thank you very much. Thank you very much for the very informative talks. I believe there are a lot of questions, but we have a separate Q&A session later, so we will go to the next speaker. Thanks, our speaker again.
All right, now let's welcome the next presenter, Professor Min-kyung Baek. She's already moderated the session in the morning, but just to briefly recap, Professor is from Seoul National University, and her research focuses on developing AI methods for predicting the structure of protein and interactions of biomolecules. Over to you, Professor Baek.
Okay, thank you for a kind introduction. And good afternoon, everyone. I'm Min-kyung Baek from Seoul National University. And it's a great honor to join this important discussion on how AI is transforming drug and medical product development. So today, I'd like to focus on AI-driven protein structure prediction and its opportunities and challenges, and more importantly, how we might think about regulation in this rapidly evolving space. Last year, the Nobel Prize in Chemistry was awarded to 3 scientists who have fundamentally changed how we think about proteins. Demis Hassabis and John Jumper from Google DeepMind, who developed AlphaFold, the breakthrough AI system for protein structure prediction, and also David Baker, our keynote speaker, who is a pioneer of computational protein design. And both groups are actually went on to found biotech companies aimed at drug development, which attracted enormous public and industry attention. And this award sent us, I think, strong signal. AI-driven structural biology is no longer just an academic pursuit. It is now viewed as a core engine for future therapeutic development. Protein structure are not just beautiful pictures. They are the foundation of modern drug discovery. So let me give you some examples. The first one, the development of protease inhibitors for HIV therapy was only possible once we knew the atomic structures of HIV protease. And structure-guided chemists to design molecules that fit precisely into the active site, blocking viral replication. Similar to the COVID-19 antivirus, the same was true during the pandemic. So the structural insights into SARS-CoV-2 main protease and the spike protein were critical for developing both small molecule drugs and also vaccines. And immune checkpoint inhibitors, understanding the interaction between PD-1 and PD-L1 at the molecular level, enabled the design of the antibodies that disrupt this checkpoint, which revolutionized the cancer therapy. And these examples all point to one message. So without the structural knowledge, many of today's breakthrough therapies would simply not exist. And this is exactly why AI-based protein structure prediction method has drawn so much excitement, because it basically promises to accelerate these kind of discoveries. So for decades, we have relied on three experimental methods to determine proteins' three-dimensional structures, which include the X-ray crystallography, nuclear magnetic resonance method, and also the cryo-EM. And these techniques have provided us with invaluable high-resolution structures and transformed the biology, but they also come with serious limitations. So they are basically very time-consuming and also costly, often taking months or years to determine just a single protein structure. And also, not all proteins can be crystallized or expressed in sufficient amount. And also the flexible regions, membrane proteins, or some complexes can still remain elusive. So because of those limitations, the protein data bank today covers only a fraction of proteins encoded in genomes. And this gap is exactly what motivated the development of computational approach. So if we can predict accurate structures directly from the protein sequence, we may bypass many of these bottlenecks and accelerate both basic biology and drug discovery. So when we look at proteins across evolutions, one striking pattern actually you can find. So let's take myoglobin and rhodopsins as examples. So their amino acid sequence differ greatly across species, but their 3D structures, as shown here, the overall fold are highly conserved. And this observation tells us, while sequences may diverge, structures still remain stable. And the reason is pretty clear. The structure determines each function, so evolution tries to maintain their function by maintaining their structures. So if structures is conserved, then where is that information hidden? So it turns out that the multiple sequence alignment, in short MSAs, which basically capture how proteins evolve across species, it contains those structural patterns. So residues that co-evolve often end up closed in 3D space. So if we can detect those structural patterns in the MSAs, then we can begin to reconstruct the underlying protein structure. And this was a kind of conceptual leap. The evolutionary information actually encode the structural rules, and what we need is just the right tools to decode them. And this is where the AI comes in. So AI basically uses hierarchies of multiple layers to learn some representations from data. And it is well known that AI is exceptionally effective at learning some hidden patterns. So when we provide this system with tons of training examples, then they begin to understand the hidden patterns and predict something useful. So if we can provide tons of protein structures in the PDB as a training data for this AI system, then this AI may understand all the structural patterns hidden in evolutionary data and may predict the 3D structures. So building on this idea, the researchers, including myself, developed some powerful AI systems that could actually predict the protein structures directly from the sequence, which named as the AlphaFold and Rosetta Fold. So now with those protein structure prediction AI, we can reach near experimental accuracy in many cases, as shown in the examples there. And this is a kind of turning point. So for the first time, we could move from a sequence to a reliable 3D models in hours, not years. And in just a few years, the field has moved beyond the proteins alone. So with Rosetta Fold or Atom and AlphaFold 3, we can now model not just proteins, but also their interactions with nucleic acid, ligand, and also other biomolecules, or in an atomistic framework. And this shift the field very significantly. Biology is not driven by isolated proteins. It is driven by the networks of interacting molecules. So if AI can help to predict these interactions at scale, the impact on drug discovery could be profound. So as soon as this method appeared, the question was, could this be a game changer for drug development? So before we ask whether AlphaFold or other AI methods can help the drug discovery, let's remind ourselves what is the current drug development pipeline looks like. So it is a basically long and difficult journey. So it typically takes 10 to 15 years from discovery to approval, and the cost can reach billions of dollars, and the overall success rate is less than 10%, I would say, meaning that vast majority of candidates actually fail somewhere along this way. And this inefficiency is exactly why new technologies attract so much attention. So if we could shorten the timeline, reduce cost, or improve success rate, even modestly, the impact would be enormous. So now, where could AI like AlphaFold realistically help, especially for these early pages from target identification to lead optimizations? So let's begin with the target identification. So one example is PD-L1, I'm showing here, a protein that became a central target in cancer immunotherapy. So understanding how PD-L1 interact with each binding partner, the PD-1, was essential for developing checkpoint inhibitors. So here is a question. Can an AI-based protein modeling method find an interaction partner for target protein to understand disease mechanism for better drug discovery? So I would say yes, partially. AI tools can sometimes predict potential interaction partners and even provide some structural models of those complexes. Then why is this possible? Because evolutionary information does not only preserve individual protein structure, it also leaves some traces of how proteins interact. So when two proteins bind each other, mutations in one partner are often compensated by correlated mutations in the other to maintain their interactions. And these co-evolutionary signals are captured inside a paired multiple sequence alignment as shown here. So what does this mean in practice? So if such patterns exist in the MSA, we can often predict whether two proteins interact with each other or not. And if the signals are strong enough, then AI models can go one step further, so they can predict the actual 3D structure of the complexes. So evolutionary history provides not just blueprint of individual structures, but also their molecular interactions, and it helps us to decode the interaction patterns. So here is another example of what this approach can do. So we basically use RosettaFold and AlphaFold to systematically model the interactions among proteins in yeast, one of our model organisms, to understand the biological process. So by applying those AI across the entire proteome, we could predict thousands of potential protein-protein interactions, and also we can propose the structural models of their complexes and reveal new biological insights into pathways and processes that had not been mapped before. So in other words, this was not just confirming known interactions, it was discovering new interactions. So after the yeast interactome study, this idea quickly spread, and many research groups began to extend AI-based protein structure prediction to tackle not only single proteins, but also their interactions. So some have developed specialized AI tools dedicated to protein-protein interaction screening, or others are applying those methods directly in large-scale interactome screening across the entire proteomes. The result is a growing wave of studies that use AI to uncover networks of molecular interactions, providing insight into disease mechanism and highlighting new potential drug targets. So what began as structural prediction is now evolving into interaction prediction at scale, a natural but very powerful next step. So what does this mean for drug discovery? So if we can use AI to predict protein-protein interaction networks and then combine them with bioinformatics analysis on disease data, we may be able to identify new drug targets that would otherwise remain hidden. So this is a vision of what becomes possible. So basically, we can map out interaction landscape, then overlaying it with disease-associated mutations or expression changes, and maybe highlighting the proteins that sit at the crossroad of the disease mechanism. So in other words, AI-based PPI prediction can help us move from sequence data to the network insights and ultimately to target identification. But what if the disease mechanism is not just about the normal protein structure? So what if it is caused by pathogenic mutation or by changes in post-translational modification profile? So here, today's AI methods actually still struggle. So they are basically trained mostly on wild-type proteins in static state in the PDB. So they are not yet able to reliably predict how a single mutation or a phosphorylation, glycosylation, or other PTM will change their structure. So in other words, when disease is driven by subtle but critical alterations, current structure prediction methods often fail to capture the consequences. And this highlighted an important limitation. So AI can give us powerful insight into normal structures and interactions, but it is not yet sufficient for modeling dynamic changes that are central to many diseases. Now, let's move to the next stage of the pipeline, the compound screening and lead optimization. So here the question is very practical. So are AlphaFold models accurate enough for molecular docking? So in other words, can we take an AI predicted structure of a protein and put small molecules in the binding pocket and trust the result enough to guide the drug discovery? This is a kind of exciting idea because if it works, it could dramatically accelerate the virtual screening. But as I will show you in the next slide, the reality is more complicated. So can we directly use the AlphaFold models for molecular docking? So the answer is not yet, not reliably. So there are two major problems. So one problem is errors around the binding pocket. So even when the overall fold is accurate, the local geometry near the ligand binding site is often off by just enough to mislead the docking algorithms. And also it's hard to consider kind of induced fit effect. So proteins are not rigid, they are very dynamic molecule, and they often undergo conformational changes upon ligand binding. And current AI models usually capture only a single static structure. So because of those issues, docking against AlphaFold models can fail to identify true hit or generate many false positives. So while AI-based structures are a huge step forward, they are not yet a drop-in replacement for experimental structures in compound screening. And how about AlphaFold 3? So recently, the AlphaFold 3 extended its scope to include not only proteins, but also their interactions with ligands and other biomolecules. So this is an exciting step forward, and early demonstrations show that it could generate complex structures directly from sequence and small molecular input. But when it comes to drug discovery, the reality is still sobering. So recent independent studies have shown that while AlphaFold 3 can sometimes capture ligand binding poses, it often lacks the accuracy and reliability needed for drug discovery. So AlphaFold 3 struggles with dynamic systems that require induced fit or conformational selection. So in many cases, the predicted structures fail to match the conformations observed experimentally. So even when we provide the antagonistic input, AlphaFold 3 consistently predicts active state GPCR conformation, which against the input small molecules function. And this shows that its training data did not adequately capture the diversity of receptor state. Also for systems like PROTAC-induced ternary complexes, AlphaFold 3 often fails to produce accurate protein-protein interfaces, limiting its utility in multi-body interaction modeling. And perhaps most importantly, the AlphaFold 3 does not give you the binding affinity. Their internal ranking scores show no correlation with experimental binding affinity, so it's a kind of structure generator, not a scoring engine. So while AlphaFold 3 is impressive as a structural tool, those limitations remind us is not yet ready to drive compound screening or lead optimization in practice. So does all this mean there is no real revolution in drug development? So not quite. The real breakthrough may lie not in predicting existing structure, but in designing entirely new proteins. So this is where the promise of de novo protein design comes in. So instead of just asking what structure does this sequence fall into, we can now ask what sequence will give us the structure or function we need. So this shift from prediction to design has the potential to transform how we think about therapeutics. So I will skip this slide because David Baker already gave a good introduction about what is the protein design. And the modern, the AI-based protein design pipeline can be thought in those 3 steps. So backbone generation with RF diffusion, and sequence design with protein MPNN, and also validation with some AI models, and also experiment. So this pipeline takes us from structural idea to the protein sequence, and finally to validated molecules using AI. So one of the clearest impacts of AI is on their success rate in protein design. So in the past, rational or random design approaches often had very low hit rate. So researchers would propose hundreds of designs, but only a handful, if any, would fall correctly or function as intended. So with the introduction of AI-based methods like RF diffusion and protein MPNN, this has changed dramatically. So now, the fraction of designs that are stable, well-folded, and functional is orders of magnitude higher. So let me close with a few key points. AI has transformed protein structure prediction. The tools like AlphaFold and RosettaFold made near experimentally accuracy accessible to the entire community. And this opened the door to new opportunities in drug discovery, from target identification to lead optimization. But it still struggles in some sense. And also beyond prediction, the generative AI enables the de novo protein design, which may open the new opportunities in therapeutic design. But these methods are not perfect. They still struggle with conformational flexibility, post-translational modification, multi-protein systems, and also affinity scoring. So experimental validation is still essential. So the message is AI-based protein modeling is better than ever before, but still falls short of what is needed for drug development. But we are still keep making the improvement to help the drug discovery. So I'd like to thank all the people who worked together with me, and I'd like to thank the organizers having me here again. Thank you.
Thank you, Professor Beck, for the excellent presentation here. Now I would like
because we have some time, but however, we have a Q&A session at the latter part of this session too, so we'll move on to the next speakers. Yeah, please.
Thank you again for bringing us back the time. And the next speaker is Dr. Bruce Church. Since he was already introduced in the morning, I would not repeat the step. Welcome, Professor Bruce. Over to you.
Hello, can everybody hear me okay? All right, so I'm going to give a slightly different example. So this is causal AI. It was nice to see us on the upswing in the chart of adaptation. So we're building digital twins, and what we mean by a digital twin, because there's a lot of different versions of it, is a deep model that describes the mechanism that gives rise to the observed behavior, the observed outcome. And we use them for drug discovery, for identifying novel targets, where we're really trying to learn the biology, not just not just looking at it as a target, but understanding how that target, how the biology connects that target up to the clinical outcomes, and also how to select patients for a given therapy, where we already know the intervention, but we would like to understand what modulates the response to that intervention. So I was trained as an engineer, and as an engineer, you want to have a circuit diagram, a model, a deep model that describes how the thing you're studying, the disease you're trying to understand, your disease you're trying to prevent, operates. And so a circuit diagram tells you how any potential intervention, what the outcome would be without running a single experiment. With this model, with all the components, when you know the entire circuit diagram, you could make a prediction about what would happen if I cut a wire or changed a resistor or changed the properties of a transistor without actually doing this. And this is how engineers work. They do causal modeling given the known model, given the known structure. And here's a similar diagram from cell replication in cancer. And it looks similar. We have edges, we have nodes, but this is more of a knowledge map. This is about what proteins talk to what proteins, what pathways are involved. And the problem with this is it's not necessarily that mathematical. There aren't equations behind those edges, but also it's incomplete. We know so little of the biology. Even if every edge, every relationship in that model were correct, and we still know so little that these models aren't useful for predicting what outcomes will be. Most of the biology, all the hard work that people have done only represents maybe 5%, maybe it's 10% now of the biology. And what we're using is causal AI to try to reverse engineer that biology at scale. So we're using causal learning. So causal AI is just causal learning at scale, where we're distinguishing between correlations. So what happens when I see something? If I'm doing differential gene expression to see what are the differences between gene expression levels in patients that have the disease and don't have disease. I'm looking at correlation between the disease and the gene expression. But what we really want to do, if we want to change things, is intervene. And we want to answer questions like, what happens when I do? And causal inference takes observational data, not the gold standard randomized clinical trial data, but observational data, and learns from patterns of correlation in that data what probable models of the causal relationships are. So does from observational data, tries to learn, does A cause B? So we do this, I can show you examples with small numbers of variables how it works, but we do it at scale, at the scales required for biology. We take DNA sequence, gene expression, proteomics, clinical EMR, patient demographics in labs, basically at least 3 layers, DNA, RNA, and clinical outcomes. We don't generate any of this data ourselves. We have partners, people who understand the diseases very well, that help guide us in what the appropriate modalities are to measure. And we take that data, we put it together with causal AI, The math behind this is all open source. A lot of it was developed by Judea Pearl in the late 90s, early 2000s. We won the Turing Award in 2011. We've married that with tools from statistical physics that allow us to do this at scale. So how does it work? In principle, it's very simple. There are 3 steps that we do. One is we take all the variables, all the biological variables in the model, all the DNA changes, all the RNA expression levels, protein abundances, clinical outcomes, and we enumerate simple models that could connect them up. Everything is an input, everything's tried as an output. We enumerate all the possibilities, and typically trillions of network fragments are scored and stored off. The hard part is where we put them together into a generative model of the biology, the optimization, where we take these fragments and we stitch them together to build a model that describes all of the data. And we use, this is where the statistical physics comes in, we use Metropolis Monte Carlo to walk the space of those models and find those that are both probable and parsimonious. And then to make sense of them, we don't just go look at the models because they're at biological scale. We do synthetic experiments on them. And because we have a distribution of models, we get a distribution of predictions that give us a self-consistent estimate of our error bars. So we know when we know and we know when we don't know. So here's an, oops, we went back. Okay. So when we, so in a biological problem, we'll pick the appropriate modalities. We put that together into a data frame with just a matrix where all the observations are the rows, the patients are the columns. We do use complete data. We found in principle that the computational effort for going higher to using incomplete data raises the costs. And we don't need that much data. We only need a couple hundred to 1,000 patients worth of complete data because of using the statistical physics methods for doing that sampling. We do the enumeration, we sample the networks, and what comes out is the digital twin is this ensemble of models that are all consistent with the data. So, for example, at the top, the triangles could be the DNA variants. The circles would be the RNA expression or protein abundances, and the diamonds, the clinical phenotypes. And since we're learning the biology ab initio up out of the data, we're not reading it out of a database. We both relearn known biology, represented by the black edges, the black arrows, but also novel biology, new connections that are suggested by the patterns of correlation in the data that we're learning from. And you can look at this diagram as a knowledge map where the arrows tell you what gene influences what other gene, or what DNA variant drives what protein abundance. But behind each of those edges is an equation and parameters that allow us to do the simulations of if we were to intervene, if we had a molecule that intervened on this protein and we knocked it down, or we increased its abundance, how would that change flow through those edges, through those models out to the clinical outcomes? So we use this in 2 ways. One, for drug discovery, where we're predicting the average causal effect of an intervention on every single variable in the model, and there might be 20,000 of them. And so on the left is an example of a good intervention, a good target, which has a causal path to the outcome. And so you can see the distribution of the outcomes result of that impact of that synthetic experiment is that it has moved. It's changed the outcome. On the right is a potential target that's not a good target. It does not have a causal path, even though it's correlated with the outcomes because of the common causes above the DNA variables. So it might show up in a differential expression as a good target, but when you model this and you model the impact of the intervention, there's no change in the outcome. The other way we look at this is due to drug simulation, and this is closer to the world that you all care about in terms of clinical development, where we're trying to identify the patients that will respond better to an intervention, biomarkers of differential response. So we can go through the entire network, given a known intervention, and ask what DNA changes or what biomarkers might predict a differential response to a particular intervention. So we have a number of validated examples where this has worked in terms of, there's an Alzheimer's disease model, and I'd love to talk more about our model versus yours over coffee later. We work in oncology, neuro. I gave a cardiovascular example last time. But today I'm going to give an example in prostate cancer. Oh, before we move on to that, so the other place we're doing digital twins is in the model systems. So we primarily build digital twins from real-world observational clinical human data, but it's also possible to build these for the model systems themselves. And we're starting with the placebo arms and model systems. It's the easiest, simplest problem to solve. And we're working our way up into standard of care, where we're seeing the same drugs applied again and again in those model systems, so we have lots of data. And we see that these models are going to feed into each other, that the information from the simpler model systems will improve the predictions made by the more complicated model systems. And ultimately, in the short run, these will allow us to decrease the number of animals that are used in experimentation, but in the long run, we'll be able to do better discovery with them. So in this prostate cancer model, this was a cohort of patients from a real-world data set, 250 prostate cancer patients. We had a whole exome data. We were looking at about 7,000 variants, both germline and somatic variants. So short variants, copy number variants at the gene level, rearrangement variants where genes were fused together, about 20,000 RNA-seq expression levels. We had the patient's history, so how did they present, what treatments were they treated with in the past, and how did they respond to those treatments. And we had current treatments that were measured around when the molecular data was collected. And the outcome for this model was overall survival. So when we went through the REST process for reverse engineering the digital twin, that space that we're looking at for building it's about the network fragments, the local pieces, the building blocks that we could imagine happening in that model. There are about 10 to the 24 of them. And when we put those together and ask how many possible models would we have to look at if we were evaluating them one by one, it's clearly more than the number of atoms in the universe. So we don't do that. This is where the sampling methods, the Metropolis Monte Carlo, comes in. We embed prior knowledge at a class level. So this diagram on the right is how we thought about the different classes of variables and the constraints between them, and that helps cut down that hypothesis space, but it's still astronomically large. What comes out is a network that has, in this case, I'm showing the average graph over about 1,000 networks. We sampled 1,000 networks to cover the diversity of models that were consistent with the data. The edges, if the less transparent the edge is, the more certain we are, the more models have that same edge in it, and those circles represent the variables. And obviously that hairball is too complicated to do anything other than show you how big the models are. But what we can do is zoom in on a neighborhood. And what I'm showing here is the average graph in the neighborhood of the androgen receptor, AR, up at the top, and OS, overall survival, at the bottom. And the edges represent what depends on what. The color tells you, is it upregulation or downregulation? So if it's blue, if the input goes up, the output on the arrow goes up. If it's red, it goes down. And so you can see things like RB1 loss, which are very common in, in prostate cancer, drives downregulation of RB1. You can see E2F1, FOXM1, TP53, very important players. And none of this was pulled from the literature. This is all learned ab initio out of the data. These are the models that are most consistent and simple that reproduce the training data. To validate these models, we do a number of things. We look for known knowns. We also try to reproduce past experiments, in this case, clinical trials that were done. These were, we looked at patients that had, that were metastatic castrate resistant and looked to see if we could recreate the trials that led to the approval of abiraterone and enzalutamide. And in both cases, the actual trial result is the red dot, and the confidence interval is the bar. That's the readout from the trial. The blue histogram is the distribution of hazard ratios predicted by our digital twin. For a counterfactual simulation, where on the first panel it's abiraterone, where we took every patient and we put them on abiraterone and looked at what their outcome was, and then we took that same patient, we did the counterfactual, and put them on an ADT, and we measured the hazard ratio. And since each model, each network in that ensemble of 1,000 models is slightly different, each model makes a slightly different prediction. That gives us our error bars. That's our confidence interval on the outcome. So in both cases, we've got the rough hazard ratio right, and we're a little bit broader, but those trials had a lot more patients than we had in our cohorts. Okay, so that's a very simple, crude, high-level cohort validation of this model, but it also has a lot of molecular detail in it. So if we look more closely around the androgen receptor, we can see, remember I said we recover known biology, and some of that is blindingly obvious. Like, for example, the AR gain up there represents a copy number gain of the androgen receptor gene. And it turns out, obviously, if you have a well-known biology, is that if you have androgen receptor gain, the androgen receptor gene expression, it's overexpressed. There's a high level of it. And you can see that over on the right. So if there's no AR gain, there's low levels of AR. With AR gain, it's much, much higher. And why that's interesting is androgen receptor overexpression is a potential, is a mechanism of resistance to enzalutamide. It's one of the ways that the cancers escape treatment with enzalutamide. Well, that's well known, that AR gain drives overexpression of AR, a known. But what wasn't known is it turned out that the model identified, the digital twin, also identified that there were two germline mutations, one in axon 2 and another in patch 1. And both of these interact with that copy number gain to supercharge the AR overexpression. And you can see that in the simulation results in the second panel, where if you have no AR gain, there's no difference in your AR expression level. The two histograms box plots are very close, whether you have wild type patch 1 or the mutant patch 1. But if you have AR gain, then whether you have this germline mutation in patch 1 matters. And if you have the mutation, you have much higher androgen receptor gene expression. So if these are germline mutations, so you're born with these, they could be measured, they were there before you had prostate cancer. And so they're potential biomarkers that could predict whether a patient might become resistant to enzalutamide. So at this point, it's interesting biology, it's just an idea that's coming out of the model. But one of the things we did is we changed the constraints we had on the model building. We said, hey, well, let's just let the germline mutations drive what's happened with past treatments. And so it turns out when we did that and built a new model, when you look at patients who have failed enzalutamide, what I mean by that is they were treated with enzalutamide and they progressed after being treated with enzalutamide. It didn't control the cancer. The most likely predictor of failing enzalutamide in a past line of therapy was having that germline patch 1 mutation. So here we have a little bit of epidemiological evidence that there's a connection between the germline patch mutation and failing enzalutamide, but also a more detailed molecular mechanism, all in the same model. So there's much more work to be done here to flesh this out, to add in more detail in those arrows. If we add more modalities, we'll be able to understand better what the mechanisms behind them are. But this is an example of novel biology recovered from a digital twin that could drive patient selection. So I'd like to stop here. Thank you.
Thank you very much, Dr. Church. And I think we're due for a coffee break. And I'm not sure, MC, are you coming?
Thank you for sharing remarkable achievements and the marvels of technology. To all the speakers and the moderators, please give another round of applause. Thank you. Now we will have a break and we will resume the session at 3:20 p.m. So all the participants, we ask you to be seated by then. And now I'd like to invite our speakers and moderators for photo session. The panels of the session 2, would you please come up to the stage for photograph? 네, 좌장님, 연사님들 무대로 모시고 기념사진 촬영하도록 하겠습니다. 세션 2 좌장님과 또 우리 연사분들 무대로 모시겠습니다. I'd like to invite the speakers and the moderators for photo session. And after second half of the session 2, we will take another photo with a good photo. So you may take a picture at that time. So would you please come up to the stage? Do you want a heart cross? You love it? No, okay. Please give a big smile. And this time we'll take a pose with the heart cross, AIRIS. And please give a big hand for the success. Would you please give a big hand? Thank you. Thank you very much. So the session will start at 3:20 p.m. I will see you soon.
제가 저기 가서 해봐도 될까요?
Ladies and gentlemen, may I have your attention, please? Now we will resume the session 2. So all the guests of honor and participants, would you please come into the hall and take your seat? Thank you very much. The moderators, would you please take the stage and welcome the speakers?
So we have 3 remaining excellent speakers. Then I'd like to introduce from the first speakers in the latter session. So the first speaker is Mr. Pat Byron, Senior Regulatory Specialist from Philips. So he's going to talk about trend and convergence, lesson learned from other fields. Please. Yeah. We already are. Yeah.
This works? Okay. All right, so yes, you have me again. This time taking and talking about some of the standards going on. So I know that we're tired, and I know that talking about standards isn't necessarily the best thing to do after a break, but we'll see how many people fall asleep. So first of all, I wanted to take and share just a personal story that many years ago I was doing research and I was going out and visiting different hospitals, you know, and trying to understand what is healthcare really like, not what the engineer thinks it's like, but what is it really like for the doctors and nurses? And so I asked, you know, the question of everyone that I ran into, which was, what's different now than it was when you first started? And she was very frustrated at the kind of time that she spent with the computer and on the computer and not taking care of patients. In fact, when I asked her, she slammed her hand on the countertop where she was at, so she was like, ooh, okay. And she's like, you know, I became a nurse to take care of patients, but I spent all of my time taking care of computers. And, right, and all that laughter, et cetera, we can completely understand that. And this is actually one of my driving factors of what I'm doing is, can we get the AI to handle some of those things and allow our caregivers more time in giving care? That's what really drives me for this. So AI has been used in many, many industries, obviously, and imaging has been using it for many, many years. But I noticed, though, when I talk to people, we're only thinking of a couple examples. So I wanted to show a couple different examples for the benefits, and as well as then talking about the standards that are going on there. So there's a lot of different uses out there, and I know, you know, some of the things that, you know, immediately come to my mind when I think about this technology and this field is, oh, it helps identify where cancer is, or where it probably isn't. But there's other benefits too. Some applications that I've seen reduce the time it takes to do the scan by using AI. It is more efficient and you know, uses less resources, and it gets the job done faster. Well, that's good. That's very, very good. Another app that I know of is that it just allows the clinician to have a conversational discussion and converting those findings into reports. And so it really helps the nurse or the doctor using the machine do it faster and do it in a way that's more explainable. I even know of some shavers and toothbrushes that have built-in motion sensors for some of this stuff to optimize it. Ultrasound also used to, you know, automate some of the positioning you need to take and do for doing some of the ultrasound. It also takes some additional measurements for it, et cetera. Obviously, this isn't everything that's going on in our industry, but I just wanted to throw out some of those. Now, switching over into the standards land, there's lots of different projects going on. I absolutely love this quote. Good judgment comes from experience, and experience comes from making mistakes and having poor judgment. There actually is a lot of disagreement on what AI is. A coworker had wanted me to throw this in of a definition from OECD defining AI. I know I was on the IMDRF AI committee. First thing was to develop a glossary. We had 16 different definitions for AI. We argued and argued and argued, and then finally came to the conclusion that, you know, even if we did settle on one, the fact that we had 16 different and we argued so much, no one's going to use it. So let's not even bother defining AI in it. So I thought that that's sort of an interesting kind of fact for this. When I'm putting together these slides, there's always so much more that I want to take and communicate to you. And so I'm not going to read, but I know with limited time, I can't read all the words on all of the slides to you. So you'll see on some of them that I bolded some of the main takeaway points that I wanted to highlight for folks. Just wanted to make sure that you know, the audience agrees that for machine learning systems, the data is defining the logic. It's not me going out and interviewing 15 different professionals, see how they decided on the diagnosis or not. Now, because that data is defining it, well, you might have problems of it being incomplete or incorrect, or there could be just a coincidence in the data that isn't relevant. I got a story for that in a second. The data that you have might not really represent the population that you need. I know of some projects where, well, the data that I could get was from a big city, and I can't get data from a 60-bed hospital that is hours from anywhere else. And well, but the patients at the tiny little hospital in the middle of nowhere aren't necessarily the same as the ones that live in downtown Chicago, you know, near me. So the data that you have might not be right. And then things change over time. I remember doing an analysis, and I found that for this one particular place that I was investigating, there was almost no reports of patients having the flu between May and September of that year. I have to giggle on that, well, because it's not winter of where I was looking at, and people usually don't get the flu during the summer. But, you know, that as an example says you really need to know what the data means that you're gathering when you're taking and programming it. Okay, now finally getting around to talk to standards, Pat. There's a big group in ISO IEC called SC42. It's developing a bunch of AI-related standards. However, they're horizontal standards. They're trying to write standards for multiple industries. In bold, I'm highlighting saying these horizontal standards, I don't think we can just grab one of those standards and say, yes, healthcare, you have to do this. Follow everything that this says, the end. It might contain ideas that we use in healthcare. It might be easier to, instead of coming up with our own definitions, take a look at what their definitions are for things, and then see if we like it or see if we want to take and tweak it. Unfortunately, I've talked to several lawmakers in different countries, and they think that whatever SC42 says applies to everybody, works for absolutely all industries. And well, no, that doesn't. There's some things we can use, but it doesn't mean that what they're writing is about that. And I have an example coming up for 23894 about risk management. So I thought it might be useful, and I know that you probably can't read this. I intentionally made it so that you can't read this, just because you can get you'll be getting the slides later, and I think having a longer list is going to be more useful than you not being able to read this in the next 30 seconds. But it just shows you the wide variety of the things that SC42 is working on. And also wanted to take and highlight some of the things that are confusion that some people have between the horizontal and vertical standards. I highlighted one that's talking about functional safety and another one talking about risk management. And for us, that's the same thing. Risk management is about people's safety. You know, pretty much, that's what it is. For them, they mean something completely different, and it's so different, in fact, that I highlighted the fact they don't even have the same number. It's not like there's a -1 for generic risks and a -2 for healthcare specific stuff. No. The problem is that lawmakers don't understand this difference between horizontal and vertical. Very much used to the standards that we follow are specific to our field. Those are called vertical standards because they just dive down into the deep parts of our particular field. I like to take and use, what SC42 is doing, take a look at some of the horizontal standards and what ideas that they have, not saying that we just adopt whatever they write, saying, ooh, they might have some good ideas, some things we might not have thought about. Let's harvest those few ideas and work them into our medical device standards. One of the things that had come up recently is some of the horizontal standards that I've heard about assume we have absolutely no quality controls whatsoever, whereas everyone I talk to in the industry follows 13485 in addition to whatever your country-specific regulations might be. Well, SC42 was assuming you had nothing, and so they it took and rewrote it. And again, the challenge is they use different words, even if it's the same concept, they use different terminology. Some of the words that we use, they have different meanings for it, and some of the things that they're doing just doesn't apply to us. And we can spend a whole lot of time trying to be compliant with these horizontal standards, and it does absolutely nothing to improve our products and improve our patient safety stuff. Unfortunately, the lawmakers that I've talked to don't understand this difference between horizontal and vertical for it, but sort of wanted to prep the audience because this is probably going to be an argument that you're going to have sometime in your future. Now, I'm not saying that horizontal standards are worthless. I harvest them all the time to see what kind of ideas people have, and then, though, I rewrite them to be specific to our industry, use the words that we use the concepts that we use, etc. Also did want to at least mention, there's a group, it's not a horizontal standard, but under TC62 that runs a lot of the standards that we follow, there's a group called the SNAG. They've gone out, taken a look at some of those horizontal standards and said, ooh, yes, this is a good one. Ooh, yes, this is a good one too, you should probably look at. So just wanted to let you know that you don't have to look at thousands and thousands of horizontal standards from other places. The folks over at SNAG had done a nice little report for you. I want to do a deep dive on 3 particular projects going on when it comes specific to our industry. So these are medical device standards that we talk about next. One, talking about AI risks. When I first started researching this stuff a couple years ago, I have a very strong passion for risk management. I do a lot of adverse event investigations in a previous job, and really passionate about risks. And as I'd be reading some of these stories, I'm like, wow, if I was the programmer, I would have made that mistake. If I was the programmer, I would have made that mistake over there too. And so I was really worried that, oh, wait a minute, there's new ways that AI can fail that I hadn't thought about before. So put together something real quick, got it published in late 2023. This is a US standard only, and it's a big catalog of different kind of risks, different kind of failure modes that you might not have thought about. I did it as a US standard just to get it done quickly and out there so people can start using it, but I've taken that to the same standards committee that owns 14971 and 24971. I've talked to them, started a project that we call 24971-2. And so that's just this big catalog over at the AMI, adding in some extra things there. And then as soon as we get that published, I'm going to go back and add in some of the generative AI failure modes. Sometimes I get questions like, well, why don't you include the generative AI now instead of making me buy this thing twice? Well, if I add the generative AI, it's going to be another year. I'd much rather something useful you can access as soon as you can, and then go back and get another one the year later. Some of the examples, this isn't everything in there, but just some of the potential problems that we can have that 24971-2 talks about. Your data has some errors, you aren't handling some of the outliers, so you have A patient, the medical record says the patient is 103 years old. Is that really true? Do they really have the patient that's that old? Or, was it someone sneezed when they typed it? So how do you handle some of those outliers? And does that really help? Is the data incomplete? It might be correct, but it doesn't really tell the full story. Is the data inconsistent? The way one hospital did something, then the patient got sick, got sent to another hospital, they did things a different way. Did the patient really change their vital signs after that, or is it just that the hospital does some different stuff? Did want to take and talk about cybersecurity. I had worked on cybersecurity standards in the past, and I was really intrigued by these. You see the one with the pig, where someone took a pig, added that noise, and then now the software originally thought it was a pig, now it says it's an airplane. Wow. And found another example where first one says, software says, yes, that's a Persian cat. They changed some pixels. I can't tell what they changed, and I've been presenting this thing for a couple years now. But the software says this is now a toaster. It is no longer a Persian cat. And it's because someone went in and they corrupted the data. They hacked the system and changed some of the pixels there. So coming up with, again, another catalog of, hey, here's things you might want to think about. We'll get that published in my country in the next 2 months or so. And then, I'm taking it internationally, take it to ISO and IEC. But it's basically just a big catalog, just kind of like how I did with the risks of cybersecurity considerations. And we actually even break it down through, let me see, do I have, nope. Break it down to different parts of the data lifecycle. My point being, someone can come in and poison the data and you get bad results. But the thing is, the data might have been poisoned when the data was first collected, or the data might have been poisoned when it was sitting at the hospital, or it might have been poisoned when it went from the hospital to your product development team, or it could have been. So there's many different places where the data could have been tampered with, and so this tries to, for all of the different security concerns, break it down in different phases of what do you need to worry about when. Again, back in my project manager days, I hated the thing that just said, here's all 5,000 things you need to do, go do them. No, I'm going to try to make these standards easier for people by saying this is what you need to do during the concept phase, this is what you need to do during data acquisition phase, et cetera. So trying to make that useful, and also the IMDRF AI lifecycle goes in there as well. Also just pushing that we're not inventing these things. We're not just sitting around the standards table saying, oh, this might happen, this might happen. These are people's real world experience, or news articles that we took and found otherwise. I had mentioned that, as soon as this is done, then I'll go back and update it with generative AI, and sometimes I get questions from people like, well, no, why don't you not bother publishing it now and wait until you have the generative AI vulnerabilities into it? Well, that's going to take more time. That's going to take, at least another year. And I think it would be much better to get something useful out to people now rather than waiting for a year. It's more paperwork, a little extra effort overall, but that way there's at least something going out. Also, as part of that project, I've been asked to have a discussion about, even for your non-AI product, how can AI be used as a cyber defense tool? So I thought that that was kind of interesting and something that needs to be explored a little more. One of the last ones that I wanted to mention was there's currently a project over in IEC TC62, which drives a lot of the standards that we have in our field. They're talking about verification and validation. What's different that you have to take and do when you're taking and testing AI systems as compared to others. And then, again, what I notice is, from my point of view, I see certain failures, but from your point of view, you might see something slightly different. You might reinforce what I've seen, but you might be something different, or someone else. And you know, I really think that it's a strong thing to get together in these different committees and share the experiences. And even if your experience is much different than others, sometimes people in those committees, I'm new to a committee, I don't want to say anything, people will think I'm stupid. No, please don't. Because those different perspectives are giving us a better feel for what the reality really is. Okay, and quote in the middle of it, I forgot to bold it, for artificial intelligence to succeed, I really think we need to improve our collective intelligence. We need to share with each other, what are some of those lessons learned? What are those failure modes? How did you do it? Oh, that sounds really logical. Oh yeah, but that really doesn't work. Share those experiences. And that kind of plea for help is the entire reason that I came here. 14-hour trip, or more than 14-hour trip, 17-hour trip from Chicago to come here, because I know that I can network with a lot more people that I normally wouldn't interact with, and you're going to have different viewpoints than what I have on some things, and that means we're all going to take and learn. And that's it, and 8 seconds left, hot damn.
Thank you very much. So we are now moving to the next speakers.
Thank you. Next, we have Camille Vidal. She's the Vice President of Regulatory Affairs for Digital at GE Healthcare.
Good afternoon. Thanks for hanging around and still being here awake for this presentation. Thanks, Pat. You did not make everybody sleep, so that was great. So my presentation today will actually focus on how we, as a manufacturer of medical device, are thinking about integrating generative AI and foundation model-based technology in our system within the existing regulatory pathways. We'll see if that works. We actually will go through a case study of an interactive segmentation tool, and we'll see how, I think we can actually wiggle it through the existing regulatory pathway. But before that, I will show you a few other examples of how AI is used in our industry. It's actually a repeat of what Pat just said. So, he was talking of MRI, here we're talking of a CT system. So we have spent, a fair amount of energy developing AI in a way that is going to make the operation of our system easier for the technologists at every stage of the exam of a patient. So from the moment the patient enters the exam room to the moment the patient exits the exam room, looking at all those steps, AI can help, at the pre-scan stage, helping position the patient appropriately on the table, selecting the right scanner parameter, during the scan, creating clearer images, accelerating a scan potentially, or reducing the dose maybe. And then post-scan, processing those images instead of manual step that the technologist may have to do to eventually send those images to radiologists. Now there are steps that AI can pre-prepare for the technologist. The technologist still have a look, make sure everything is okay, and then can send to radiologist. The whole goal here is to make the experience seamless for the technologist, seamless for the patient, and everybody is happier. Another example here is an example of an AI guidance system, where on a portable ultrasound system, you now have the capacity from the system to essentially give very specific guidance on how to position the probe, how to turn the probe, or modify the location of the probe so that you can acquire a cardiac exam with this ultrasound system. Here the goal is to, by enabling this AI guidance step by step, is really to help users that are less trained at acquiring this type of images to actually capture diagnostic quality cardiac images, and therefore open up different care pathways, because now you can give to a registered nurse or to a primary care physician this tool, and maybe if they feel it's the right thing to do for their patient, they can go ahead and acquire the scan in their office. So now going into our case study. So the example I was just showing you, they were all based on deep learning. So technology that has been developed now for a little while, at the turn of the century when machine learning was still called machine learning, and then in 2010, 15 when it became deep learning. That's the technology that those those examples is based on. So what is deep learning? It's narrow data set, so data set that are very specific, one modality, and usually targeting one application, one specific task. But there is a leap forward, and we all see it in everything we do every day at work or at home. Now there is a leap forward where instead of doing task by task model creation, there are those foundation model being the basis of the generative AI, right? The GPT in the chat GPT. Those foundation models are built on massive data sets, potentially generally multi-model data sets, and that have as a goal to be applicable to a broad set of use cases. And so that changes a little bit how you will develop this type of functionality for your system, where we used to collect label data, create a machine learning model from scratch, sort of way of saying, so that you can solve for a specific task. Now you can actually take the problem in a different way, and instead of creating one model for each task that you want to solve, which there can be many, you now create a foundation model from a large set of data, and then you can fine-tune that foundation model to solve a number of different tasks much faster than having to recreate for every single task a model from scratch. At least that's the theory. So let's take an example. So a little case study that we did. So we were interested in creating an interactive segmentation tool for ultrasound. Something where the user clicks on the part of the image that they want to segment, and it will automatically figure out what is it that the user wants to segment, what's the object in this image. So our data scientists said, well, turns out Meta has created a model, it's called Segment Anything model, that is actually specifically intended to segment objects in natural images. So the way it works is that, from a user perspective, the way it works is that you present your image, you click, for instance, on the scissors, and it presents you the most plausible segmentation of those scissors. So we tried that on ultrasound images, not really knowing if there was any ultrasound images in the training data set. As it turns out, that's the contour in red that you can see in some of those images. It didn't work very well because the nature of the noise, the fact that the image is conic in an ultrasound image, really threw off the model, and the model just didn't operate as one expected. You could try to put more input, like more clicks on this image, but still you couldn't get a very clean segmentation in a way. So then data scientists used transfer learning to fine-tune the model using 200,000 ultrasound segmentation masks, and then created this prompt-based segmentation tool, which you see the result in the slide here on the green contour. So not perfect. We're talking, this is a case study. Not perfect, but much closer than what the SAM algorithm itself was able to do. So a demonstration here that, this fine-tuning process or transfer learning and fine-tuning process has some possibility of working. So how does that work? If I want to now integrate this function into a software medical device. Well, as manufacturer of a software medical device, there are a certain number of steps, and that's just a short list of them. It's not meant to be the whole list, but just, high level. First, you need to decide what's the intended use, what's the benefit-risk profile that will come from the intended use, and the technological characteristic of your device or the functionality that you're trying to build. And that will typically define what is your regulatory pathway. Then you have all the step of creation of your product and establishing the safety and performance of your product. So there is design, risk control, algorithm qualification, if you have an algorithm in there, software testing, maybe the normal software testing, V&V, labeling transparency, usability, clinical validation, and there are other things, but that's for the short list. And then, once you have established that, convince the regulator that your product is safe and effective, got regulatory authorization to go onto the market, then once the product is on the market, you have to deal with post-market controls, cybersecurity updates, lifecycle management. All these aspects are true for any software medical device, as long as use an algorithm. And AI will essentially, you include AI into your product and you have the same type of activities that need to get done. The how you will go through those activities might differ a little bit, but the activity still remains. So what I wanted to do right now is to focus on 4 points, because we won't have the time to go through everything, but focus on 4 points that raise questions when you're dealing with Gen AI or foundation model-enabled medical device. So the first one is defining the intended use. So in the example that we had, the goal was to have a fairly generic interactive segmentation tool that could be used on any ultrasound images, whichever the probe, whichever the system, and on any anatomy. And so there the question is, okay, so you have a potentially a bit wider intended use than a typical medical device, so how do you deal with that? Well, the reality is that most likely you start where you start, right? You start with the data you have on hand and the evidence that you have on hand, and that will essentially define what is the scope of your first regulatory authorization. So that's one aspect, right? You can't really go around without defining an intended use. Some folks say, well, you know, generative AI is so generic in use, I really can't create an intended use for that. Well, I would say you have to if you want to demonstrate that it's safe and effective, because you can only show that something is safe and effective at a specific set of tasks or a specific set of activities. And so the intended use, you can't skip over that. I debated it with some colleague, and we all ended up on the same page. You need an intended use. So, okay, you start with this limited intended use, but since this foundation model, you were really hoping to have a little bit more flexibility than just a very limited set of organs. And that's where this new regulatory tool available in several jurisdictions that is called the predetermined change control plan can become handy. Because if in your first submission, you're able to demonstrate the safety and effectiveness for an initial scope and present plans for making changes that will expand the use case of your system to maybe the full scope of what you were hoping your device to be used for, then you actually gain that flexibility again. All right, so that was for intended use. Next step, and Pat talked about that a lot, is the adoption of a robust risk management process. So in medical device, it starts always the same way. You need to identify and mitigate the risk. So we follow a strict process. We have rigorous, long list of things that could go wrong that we have to go through, consider them, assess the risk, and design control in our product. And we continue to do that throughout the life cycle of the product. So take Gen AI, and there are quite a few things that can go wrong. I listed a few. They are not, it's not an exhaustive list, but the famous hallucination, misrepresentation of clinical information, contaminated data, data concept drift, or over-reliance of the user on the output. Those are just examples. Well, you have to go through that process of figuring out how are you going to mitigate those different risks. And here, they're not lining up necessarily because one risk goes to a specific mitigation across, but it's just example of mitigation that you can build in your system, or calling for human oversight is probably more in the labeling, but the mitigation that you have to put in place in the development of your system so that you can, by design, mitigate the risks that come with this technology. So I'm looking forward to the generative AI version of your standard PAT, because then it's going to be much easier. But in the meantime, just have to use our brain and the best experience that we can have. So now assessing safety and effectiveness. We are back a little bit to this question of what's the intended use and what's the scope of the testing that we need to do. So here in the example, again, of this interactive segmentation tool, we wanted to be able to say it's usable on any anatomy. So we sampled different type of anatomy, and the radial graph that you see on the left represent the number of clicks it takes to get a segmentation that is 90% correct. And so, well, you can't see because it's too small, but of course our system is the best. So our system, our research tool is the best, and it's, the red central contour shows you that you need less click to reach 90% than the lime color contour that was the SAM. The other one are other example of model that can be used. And all of that is in a reference that you can find at the bottom of the slide. So I invite you to go read the paper, and all this information is in there. So, all right, so your demonstration of accuracy, robustness, and minimization of bias will probably use an independent representative data set across the set of representative system acquisition techniques and anatomical region. But that might not be enough for the full intended use that we just discussed. It will probably be enough for the partial coverage of the initial use of your system. And there again, the PCCP, or maybe post-market monitoring, will help you expand the use case of your system to what you originally wanted to reach. So that brings us to the fourth point, establishing control and lifecycle management. So there is a little bit of a belief that when you use a foundation model, it learns as you use it. That's actually not true. Foundation models are frozen unless you decide to take them and iterate on them. So that means that as a manufacturer, we need to maintain version control. So that means if you use the foundation model of someone else, you may not have full version control. Something to think of. And you need to maintain end-to-end validation. There are ways, in those foundation model, in those generative model, to increase the consistency of the output. So that's probably something that, as a manufacturer of medical device, you want to consider, since consistent output is useful in our industry. Then the aspect of adaptability and speed. I already spoke to the PCCP process, a process that is kind of crucial in that space if you want to maintain the benefit of using this type of technology. And then the post-market monitoring. A lot of those technology is built on cloud. And that's really an opportunity here as you build those new systems to really evaluate the best practice from the software industry and try to capture feedback during use of the system. In some situations, the device risk level might require that the post-market monitoring is actually one more risk mitigation in the design of your system. So all that to say that, at least in the use case that we were considering, it sounds like we are able to, in this case study, we are able to envision the inclusion of a Gen AI or foundation model-based functionality into a medical device within the existing medical device regulatory framework. And I wanted to highlight that because it's very tempting when a new technology comes around to say, oh, the regulatory framework is not going to work, we need to change it. Throw the cards and start again from scratch. Well, in fact, people were saying that at the beginning of deep learning explosion. But as it turns out, 1,000 devices or more in the US have been authorized on the market. So it seems like it actually works. So it sounds like it could work also for the Gen AI or foundation model-based medical device function. Maybe not all of them, but for the one I was presenting. There will be some adaptation that probably will be needed, right? This ability to do predetermined change control plans is something we can do in the US, in some of the region, but not everywhere. So maybe something to consider. And then, this aspect of continuous post-market monitoring for Gen AI might be necessary if you need to bridge that gap between pre-market assessment and real-world performance. Or, if the risk level of the device really justifies it. And then just wanted to finish with two thoughts. Based on my experience of looking at AI regulation across the world, as you see it on the slide, right? So we have different approach in different countries. In some country, the regulation is cross-sector regulation and will apply to our vertical of medical device. In some countries, the regulation is vertical and is dealt with by the medical device regulator directly. From a perspective of a medical device manufacturer, it's much easier when the medical device regulator is the one in charge of figuring out what is it that I need to do for a Gen AI or for an AI-based system. Why? Because they understand the regulatory framework. It's easier to get those new expectations and integrate them in the existing framework, while cross-sector regulation can create incompatibilities, redundancy, and frankly, confusion as to what applies to medical device or not. So if I have a choice, which I don't always have a choice, rarely in fact, I would prefer regulation to be vertical. Like if there is extra requirement for AI, be in the context of the already existing medical device regulation. And then finally, echoing some of my colleagues, there is a plea for international regulatory coordination because it's vital for a product that we want to bring to all patients to have the ability to enter the market with similar requirements around the globe. I'm personally encouraged when I see the great work that is done in INDRF, where experts from different countries come together and really try to align their position and their policies on how to regulate those devices. I'm also encouraged when I see the work done in a standard world. Once those standards become official, we'll have a great set of common tools and methods that we can leverage. So really looking forward to all the great work that Pat is going to do for us. But anyway, all that to say that I'm an AI enthusiast, but I do think that we need to be responsible in how we bring this technology on our medical device. Because after all, behind every regulation, every product, and every decision, there is a patient. And sometimes, as a colleague of mine reminded me, sometimes the patient is you. Thank you.
Thank you, Camille. Now I think we have one last presenter today.
So let me introduce our last speaker, Ung Bae. Mr. Ung Bae is a co-founder and CEO of Sunbit AI, leading the development of AIRIS XR, a generative AI tool that accelerates chest X-ray interpretation. He served as a chief healthcare official at Kakao Brain, and led R&D at Vuno, deriving innovation in medical imaging and biosignal AI. He is co-author of a 2025 radiology study that validates clinical value of generative AI in radiology reporting. The title he is going to present is Generative AI in Medicine, Trend, Evidence, and the Sound Boundary. Please, yeah.
Okay, thank you for the introduction, and thank you for being here today at this event. I'm the CEO of Sunbit.ai. Today, I will be speaking on the topic, Generative AI in Medicine, Service and Evidence and the SAMD Boundaries. I understand this can be a complex and serious topic. Yes. Before I begin, I wanted to provide some context about my perspective. Sunbit.ai is a startup that developed a specialized generative AI software for medical imaging. However, I'm not a regulatory expert. Therefore, my presentation today will offer insight from the viewpoint of a medical AI researcher. So I ask you to please keep this in mind as I share my thought. Okay. Here is the agenda for my presentation. First, I will introduce the latest service in generative AI-based medical LLM and discuss their clinical evidence. I will cover 3 cases, open evidence, and Professor Balmed, and our own solution, AIRIS CXR. Second, I will raise some point for discussion regarding generative AI-based SAMD. As we all know, the fundamental purpose of a medical device regulation is to ensure a baseline of safety for patient and the public. This principle of patient safety first is the foundation for our entire discussion. Let's begin with our first case study, open evidence. As you can see in the image on the bottom right, open evidence function like a search engine for medical knowledge, like Google. Clinician can ask questions and receive answers supported by evidence. Its key feature is that it's partnered with leading institutions like the publisher of NEJM and JAMA. Through this partnership, it accesses the latest information and provides clear reference for its answers. Because of its reliable data and well-cited references, it's reportedly used by over 40% of physicians in the United States. It's awesome. It is important to note, however, that this product is not a sorry for that. Yes, it's not regulated as a SAMD. Okay, now let's look at the clinical evidence for open evidence. A small-scale study was published this year. In this study, 5 physicians evaluate the answers provided by open evidence for 5 common chronic patient conditions found in primary care. Okay. The physician ranked the response on a 4-point scale across several categories, including clarity, relevance, and evidence support and decision-making impact. The results are quite interesting. The evidence support category received a high average score of 3.35. However, the decision-making impact scored much lower at 1.95. This suggests that while the tool provides high-quality support that can improve workflow efficiency, its direct influence on a doctor's final clinical decision is limited. It helps them work better, but it doesn't make the decision for them. Of course, this much acknowledge, we must acknowledge that the reliability of this study is limited due to the very small number of the test cases, just 5. Next, I'd like to introduce Professor Balmed. This is a medical co-pilot chatbot designed for doctors. The company built its own database of 2.5 million documents from medical guidelines and PubMed. The service uses a technique called RAG, Retrieval Augmented Generation, with a GPT-4 model to search this large database and provide information. A key point here is that the quality of the service is highly dependent on the quality of its own 2.5 million documents. Unlike open evidence, this device has received CE certification in Europe as a SAMD Class 2B device. As a certified medical device, Professor Balmed went through various validation processes. First, the developer built a test data set of about 900 medical queries. Then they conducted a 3-stage evaluation. The first stage was an internal test to evaluate the consistency through multiple Q&A sessions. The second stage involved testing the Q&A with specialists from various medical fields. Finally, the third stage was the test in a real-world setting with medical staff from diverse backgrounds. This is the result led to a safety index of 0.26, which supported the conclusion that the service provides safe and reliable results. Finally, I would like to introduce our company product, AIRIS CXR. This is generative AI that creates preliminary report for chest X-ray images. It was trained on a data set of 14 million chest X-ray and report pairs. And we are proud to say that it is the first of its kind in South Korea to receive IND approval for clinical trial as generative AI SAMD, which is currently in progress. And the intended user is a radiologist. The input is a frontal X-ray images, and the output, as you can see in the image, is a preliminary report, radiology report covering a wide range of findings that radiologists review. To demonstrate the clinical evidence for AIRIS CXR, I will introduce two studies that was published in the journal Radiology in this year. First, we conducted a quantitative analysis. This diagram explains how we transformed the AI generative pre-text radiology report into a multi-classific task. This was to determine the presence of certain specific findings. To do this, we developed an automated labeling model called the CheckGPT. It was trained using pseudo label generated by GPT-4 here. The full detail of CheckGPT are available on our company's GitHub and paper. And the reference standard for this test was established through the independent annotation of 3 radiologists for each case here. Result. First, the AI generative report demonstrated good performance in detecting chest X-ray findings where all 3 radiologists were in full consensus, as you can see. However, would evaluation based on classification quantitative metric is sufficient? I don't think so. So, in one case, the AI accurately detected the left clavicle fracture there, which was missed by both radiologists and GPT-4V. However, the AI report including hallucination information, it is so critical, mentioning findings since on a CT scan. However, despite no CT scan being provided. So, as in the example I just showed you, the classification metric alone cannot fully evaluate the quality of the natural language preliminary report. Therefore, we also conducted a qualitative analysis by comparing AI generative report with those written by radiologists and GPT-4. And we used 4 different metrics to comprehensively evaluate the qualitative performance here. Acceptability, agreement score, and quality score and ranking. And let me briefly explain these 4 metrics. Acceptability measures whether a radiologist would approve the report as their own without any modification. The agreement score was based on an expanded Radfield score. It's so important, and this evaluating discrepancy on a 5-point scale. The quality score uses a 5-point Likert scale to subjectively assess the report's overall quality. Finally, ranking allows radiologists to directly compare the report from our AI, GPT-4V, and human radiologists. The result of qualitative analysis was very encouraging. AI generative report has slightly lower acceptability rate than radiologist report. This is from 2 years ago. And however, our AI report scored higher on both agreement and quality compared to the radiologist report. Finally, to confirm the performance with and without our AI tool, we perform a multi-leader, multi-case leader study in 2 sessions. 5 radiologists who were leaders independently interpret the CXR blind clinical information. In the first session, they reported the CXR without AI. In the second session, they used the AI generative preliminary report with the option to revise. There are 4 weeks washout period between the first and second session. The sequence of CXR was randomized to minimize bias. For qualitative evaluation of report, 2 thoracic radiologists who were evaluated separately evaluate the agreement and quality of report from both sessions. The results were clear. The introduction of AI generative report significantly reduced average reading time about 42% for the radiologist. Furthermore, the agreement and quality score both generally improved with AI use. So far, I have discussed 3 different services: AIRIS CXR, Professor Balmed, and Open Evidence. Despite their difference, they all share the same fundamental intended use, I think. They are designed to be an assistive AI in a clinician-in-the-loop framework. Their purpose is not to replace clinical decision-making, but to enhance workflow efficiency and quality. From a physician's perspective, I think many physicians would give higher score to AIRIS CXR and Balmed than to Open Evidence, maybe. Because these two are supported by good peer-reviewed clinical evidence, which help reduce the risk of automation and cognitive bias, thereby lowering the likelihood their mistake. In addition, all of these tools allow physicians to directly assess the accuracy of the AI output. With AIRIS CXR, radiologists can verify the result against just they see the image. With Professor Balmed and Open Evidence, physicians can check the answer using their own knowledge or by consulting reference. Now, what about patient perspective? For most patients, I think if they were to use this service directly, it would be difficult to verify the result from Professor Balmed and Open Evidence due to the lack of their medical knowledge. And if they knew their physician was using one of these AI tools, they would feel comfortable with any of them, I think. In fact, most of them would not really mind. This is because their trust is strongly placed in the human doctor, not in the AI tool themselves. As I mentioned earlier, the purpose of medical device regulation is to ensure at least a minimal level patient safety. Currently, regulators determine whether a product is a medical device and its appropriate class by considering the intended purpose to assess the level of clinical significance. Here, I wanted to emphasize the difference based on intended user. A radiologist can adjust the output with high accuracy. A general physician maybe can clearly evaluate findings like a large nodule or a large pneumothorax, but may struggle with a small nodule. For patient, unfortunately, a chest X-ray might just look like a black and white image. As this shows, we already know that the clinical significant risk of the same information changes depending on the expertise of the intended user. Therefore, I would like to propose my simple idea for discussion. When we regulate generative medical assistive AI, perhaps we should also consider the intended user's ability to evaluate the AI output. As shown in the diagram, as the intended user's ability to evaluate the AI increased, the clinical significant risk of the same product could be seen as lower, like this. And this is because the expert user can, as a strong safety filter or gather. Considering this factor could help us make more nuanced decisions about whether a tool should be a medical device, and if so, and what classification level. This could be a factor in fostering innovation while ensuring patient safety. Okay. Thank you for your time and attention.
Thank you very much for sharing your experiences and very important perspective on generative AI. So now we are going to move on to the Q&A sessions. The speakers are supposed to come to the podium after the seats are set up. So, and we are going to have a 20-minute Q&A, so please be seated, audience, please. Okay, because our times are limited, so questions are given by moderators for both of us, but in case we have some time left, maybe there are some more questions from the audience. But let me start from Dr. Tala Fakuru. So yeah, thank you very much for sharing your experiences. You are shaping FDA's first AI guidances. The question is, based on your experiences, what lessons are most relevant for the global harmonization in this regulatory process? Can you comment on that? Yeah.
Yeah, so one of the most critical things, I think, to advance AI and regulatory science around AI is harmonizing glossary terms. For example, today we heard a lot about things like digital twins, but if you ask 10 people in the audience what a digital twin is, you'll get 10 different answers. FDA developed an AI and digital health glossary that I think is a good starting point. It's an evolving glossary. I know that they're collaborating with EMA on identifying common terms that the two regulators can agree on, but this might be a good place for ICH to also start with considering harmonized terms, and then taking the existing reflection papers, discussion papers, and guidance documents and try to find common elements to harmonize on so that we globally can be on the same page.
Okay, thank you.
The next question, Professor Beck. So your presentation is very technical and basic science. So the question now I would ask is more around how would you, if we pivot to the regulator's perspective, when do you think, when AI is applied in the process of building models, basically in the research to product pipeline, when should regulators step in?
Oh, that's very good question. So there are some, concerns about misuse of the protein structure prediction and protein design AI, something like to develop biochemical weapons kind of thing.
Yeah.
But I would say the product designed by AI is a virtual thing unless it's realized through the gene, expression. So I think using the AI itself is not a real thing. To it become a real thing, then, especially for the molecular works, it should be realized through the, gene synthesis and gene expression, all of those when left the validation step. So I think the regulatory system can step in those phases so that whether to check whether this gene synthesis is requested for what purpose kind of thing. Then in that way, we can we can prevent misuse of those AI, the protein design or protein structure prediction AIs to further some bad purpose, yeah.
Thank you.
Okay, let me ask a question to Dr. Hao-Yang Lee. So, Edge has been developing Edge Excellent for many, many years and is one of the pioneers in Korea in LLM and generative AI in general purposes. And based on your experiences in this development, when you use for the clinical application, maybe you can think about some specialized AI versus these kinds of foundational AIs as well. What's the pros and cons from your industry perspective on utilizing two aspects? And the second part of the question is then, based on that, you need to actually think about the regulation and getting approval from for example, KFDA. In that case, then is any kinds of difficulty to meeting the stringent safety and regulatory expectation into form of the AIs? Yeah.
So answer the first question first. When we build a general AI model, there are huge amount of open data sets out there. So there is no problem to get the data and then train the model. Large scale of the GPU infrastructure is already there. So it's very easy to build a successful AI model. But when we move into the specific domain, especially in the bio and the healthcare sector, it is really hard to get the clean data set, transparent and ethically and safely collected data set. So the technology is already ready and the infrastructure is already there. but the number of data set is way less than the AI, training the general AI model. And the second question is the regulations. Once we started to build our own the AI model, which can help the clinical trial more efficient and effective that I presented, the first challenge is in Korea, it's almost impossible to get the right data set. We can actually buy a lot of the pathology image data from Samsung Medical Center or Seoul, the hospital, national hospital. But in gene sequencing or the DNA sequencing data is categorized as private data. So private data protection is almost impossible to get in Korean country. So we try to reach the US, the hospitals such as the University of Pittsburgh Medical Center and the Vanderbilt Hospital. One of the marvelous things is in the United States, regulation is very reasonable. So if you would like to submit what kind of the purpose you would like to use this data, and once it is anonymized, you can buy the right amount of ethically and safely collected data and put those things in the trained AI model. It's very reasonable. So that's exactly why we are collaborating with a lot of US hospitals rather than Korean hospital.
Thank you.
Next question to Bruce. So you've introduced a digital twins work at Causal AI for drug discovery. Just wondering, what are because you're still in the upward stage, so what are the challenges for you to diffuse and scale? And what kind of international cooperations you would expect just to accelerate the diffusion of this?
Well, so I think validation is probably clear case studies where this has worked and where we have the experimental data. So for us, we've been able to get access to good, high quality data, especially from patient foundations, and for around specific diseases. So for example, we have a digital twin in Huntington's disease, we have digital twins in Alzheimer's, and we've worked with the foundations to gather that data. The place where we haven't been able to be as clear and as transparent is on the validation side, because traditionally our commercial partners, the biopharmas, have been the ones who are doing the experimental work. And so that is, to me, that's the bottleneck for wider adoption of causal AI, is those very clear, solid proof points. And while we can do the data sharing and some of the initial interesting biology experiments, like interesting findings, and share that with the foundations to help, with the ultimate aim of helping patients with those diseases, and that's a very clear path forward. The validation work is expensive and often is done with commercial partners. And so it takes a long time for that validation to get out. And so I think that's really where we're limited, is in that ability to show those clear case studies, because of the way that work is done in collaboration with commercial partners.
Thank you.
Okay, let me ask a question to Pat. So yeah, I think it's really great to hear there is two kinds of standardization, vertical and horizontal aspect. But my question is that, for example, even with those kinds of two categories, compared to like other standardization, in, for example, wireless communication, if you don't follow the standard, you cannot make any product and sell any product, right? But in this kinds of AI product in healthcare stuff, then what is your thought on enforcing those kinds of, enforcing standards in product? Is there any way to do the similar thing in communications, various communication?
So fortunately, I've been able to get the regulators to actually be on the standards committees and contribute to it. And actually some of it, like the cybersecurity project that I talked about, where it's a list of different cybersecurity vulnerabilities you might not have thought of for AI, I wasn't planning on doing that for 2 more years, but I had the FDA call me and say, no, do this now.
Okay.
And so I do like to treat the regulators as customers for these standards. Okay, they have unmet needs, just like our design teams have unmet needs. So let's come up with a standard and collaborate with them to say, what does good look like? And that has worked. In many ways, that's worked for me. I still, sometimes I'll describe myself as still being an engineer. I'm doing new product development or maintaining existing products because there are unmet user needs out there that the regulators have. And the more that the industry can understand what the regulators need to do their job, and the more the regulators understand what the development teams need to do our jobs, okay, this helps out everybody. And personal story, I had a very deep understanding of the value of quality systems and standards when I went to go visit my mother in the hospital and she was on one of my devices. Okay, you really get an appreciation for the value for some of those things. And again, this is why I come to meetings like this, because I'm going to run into people that I never would have run into before. You have different opinions, you have different experiences, completely open to that, and because we're all patients eventually, and we're working toward a common goal. And I try to see what those needs are for them.
Okay, thank you.
Now I have a question for Camille. So you mentioned that from GE Healthcare's case point of view, you presented a case study that is about how you actually develop a solution that's within the current regulatory pathway. But at the same time, do you see there's any need for the regulatory framework for all of the emerging data-enabled technologies to be regulated? Do you foresee any needs for innovation at some point?
So I think, I see 2 categories of those data-driven methods that exist. There is the one that is really targeted to solving a specific problem. And as far as I have seen, those targeted solutions follow this same process. You need to decide what they are going to be used for. You need to understand what are the risks. You need to validate them before you introduce them into clinical practice. And if you want to do investigation, you do clinical trial with them. So I think so in that context, I strongly believe the existing regulatory framework will remain, and maybe there will be adaptation. Maybe, on the post-market side, there will be more adaptation on that side. Maybe there'll be a slight rebalancing of pre-market and post-market activities, if that's possible. Now on use cases that are a lot more generic, where we say, okay, here is a model and use it as you see fit. I frankly don't know how we will regulate those things, or at least it's probable that the current existing framework won't work for that. But I don't know if we will get to that point of saying, okay, here is, ChatGPT version whatever, and now as a doctor you can use it for whatever you see fit. So.
Yeah, I mean, I do want to follow up, but I realize we also don't running out of time, so I'll pass on the mic back to Professor Ye.
Yeah, so let me ask a question to Bae. So yeah, actually it was a very impressive presentation, especially sharing your academic, your achievement in clinical trials and etc. And also, so I think I can appreciate your effort to get a first company to try to get the approval in generative AI in Korea. So that because of that, I have a question. So maybe perhaps you may have some kinds of experience you want to share how to collaborate with the MFDS to make these kinds of approval more smooth. So maybe can you share those kinds of experiences and what's actually what's the good feedback from the government and what helps you to develop the product? Yeah, please.
Okay. Actually, I think it was very hard time for our company. However, our MFDS helped us to acquire IND approval. So I think MFDS is so helpful and so very nice institution for medical AI company in South Korea. So our company have a plan to explore our expand our product to abroad. At the time, this clinical evidence which make in South Korea from with MFDS, I hope to this clinical evidence will be helpful to acquire the other companies' approval, such as FDA or CE or yes, PMDA. Yes, thank you very much.
Okay, great, thank you. So I think our time's up. So maybe
We need to make a quick summary.
yeah, maybe I think we need to conclude this session unless there is some like a burning question from the floor. Otherwise, yeah, thank again for all the speakers and also audience to standing up till late in the afternoon. Okay, thank you.
Thank you, everyone.
Thank you for enlightening and eye-opening presentation. We are so much amazed by the cases. Please give another round of applause. You may return to your seat. Thank you. So this concludes the official program of the first day, and thank you for staying with us until the very end. Tomorrow's program will feature 2 sessions, AI regulatory frameworks, best practices, and lesson learned, and AI technology and regulatory status at each stage of full life cycle of a medical product. So we look forward to your continued interest and participation. Thank you.